Priyanka, are we starting with the safety? Okay, before we start, we need to do a safety announcement for those who don't know the exits are. Okay, over to people at the booth.
Welcome to Hingston Hall Conference Centre. Please can we have your attention for a short briefing. There are no fire drills planned today, so if the fire alarm sounds, please make your way calmly to the nearest exit. If you think you may need assistance when evacuating, please let a member of the venue staff know as soon as possible, just in case. Smoking is prohibited throughout our site, apart from at designated smoking areas. Please ask at reception for details of the nearest designated area. Please do not smoke outside the entrance. Lastly, if you have registered any dietary requirements or allergies, please make a member of staff aware, as food may have been prepared specially for you. Thank you for listening, and we hope you enjoy the event.
Okay, I think that's me. When I get some slides. Yay. Okay, welcome to Explainable AI 24, in biology. This is a hybrid conference, so there's a lot of people online and more people come drifting in as we go on. We would like to start by thanking the people who put this together in a very, very, very short time with very limited constraints and a slightly limited budget as well. Some of them are around, you'll see them walking around. Thank you very much in advance. I'd also like to particularly embarrass Priyanka here and say thank you very much to her. She will look shamefully. She did a lot of the logistics on this. Obviously, there are a lot of other workshops that happened on during the week. One of the main areas for us was actually to try and make this more interactive. We were trying to get more workshops and tutorials together. We were very lucky to get tutorials on sequence classification, fine-tuning, and even sort of building machine learning models or neural nets from first principles. So it's a nice thing. You obviously all missed it. It is online as well. And of course, I'd like to thank the speakers as well. Unfortunately, Chris and Deogratias had travel problems, so they'll be joining us online as well. So, to try and make this a little bit more interesting, we're going to have a quick quiz, because someone said in the morning everyone's going to be done. So, how many people in total do you think are attending this conference? Although this is 50 out at the moment. Okay, how about this? I'll offer a prize of a pristine, hardly used coffee cup. Someone yell a number, a letter. Oh. No! Yay! Who did that? All right. I can't throw this because I'll hit him. Could you just pass it back to him? It's a lovely, lovely, lovely coffee cup. Yay! And how many, and look, this one is new. And it has a lid on the floor. Okay. How many organizations are participating today? A, B, C, or D? Coffee cup? Do you know, I don't actually know. C! Anybody say C, or do I get to keep the coffee cup? All right, I'll get to keep it. So we're very proud of that. We've got a large number of people. It's over Zoom, so it's geographically dispersed, and so are speakers, of course, and we have a large number of speakers from across the spectrum. So one of the things I wanted to get over here is for those of you who have keen eyes will have noticed the logo down here, BioDev Network. This is something the BioDev Network put together. The BioDev Network is something that the Sanger started in collaboration with EBI and other organizations like the CRIC joined as well. It's a community. So after COVID, there was less community activities, obviously, so we sort of started a new one, and we called it the BioDev Network. The main aim of the BioDev Network is about collaboration, because we're thinking if we're going to build these things from scratch, what do we really want from these sort of network events? And that is collaboration. That's why we had all the tutorials before this conference, and hopefully throughout this, and over Zoom, we'll get more collaboration events as well. The BioDev Network is basically a community, those who can't read, that supports learning and career growth in bioinformatics, AI, and software development, because those were three areas we thought were important for us in sort of like what we call the informatics community. The main area is it focuses on helping early career professionals connect with each other. That's what it's all about. I'll give you details of this. Like I said, it's got 11 different organizations involved already. We have a series of activities that go on. And we focus, like I said, in these main areas. So if they're of interest, please join the activities. They're all free. A lot of them will be online as well. We have the artificial intelligence community, which basically organized all this. And, well, you can guess what it does by the names. We have the software development, particularly in biology, so very RSE sort of like derived. And we have scalable bioinformatics, very software. We also, because of the way we run it, it's up to the groups in the BioDev community, the sort of areas they want to expand in. So there's been a new community set up, which is equity and tech. The sort of activities they're involved in are 3 different areas. There's community engagement, and this is an example of one of them, where we run things like hackathons and training. There's global expert visits, which I suppose this is part of, and we're going to start some special interest groups as well. Again, it's not necessarily, it's like I said, it's open to everybody, so if you're interested, please come along. There's also, because it's for early career, there's also a focus on skills development. So this is developing things like competency frameworks and try and unify them across different institutes. You know, what do you need to get better at in these different disciplines? There's going to be an online learning library, which is a work in progress. And there has been some pilots of some mentorship programs, basically to match skills to need to a certain extent, to give people experience in methodology they wouldn't necessarily get, and they bring new skills in. And of course, there's public engagement. So the activity here is Coding for Kids. It's the main one, started, as well as public lectures and scientists' cafes as well. So, I haven't got long. What do you really want to get from this conference? Like I said, this is a BioDev conference. So we're very much into the community bit. So ideas to work together, collaboration, that's really what we're trying to get out of this. Get more talking, get more community engagement online and offline. Obviously, this is in an area where the Sanger decided to invest because we feel it's an important area we need to get better at as a scientific community. So that's me doing a very lightning introduction for the BioDev network, which I hopefully have advertised correctly. Please, if you're interested, join up and we'll provide more information. So that's an introduction to the conference. The thank you is to all the organisers and also to the Wellcome Sanger Institute for funding all this, which is lovely. So what I'll do now is I'll introduce Tim, who's going to give the first talk, I believe. So Tim is the director of ELIXIR, which most of you should know. He's currently also, I believe, Professor of Bioinformatics at King's still. I probably should have checked that. No, he's not. Yes. Yes, he is. But obviously we know Tim from years and years because he used to be head of informatics at the Sanger. We know he's involved in TREs and other sort of things as well. So nice background across the board. And most of us in the room know him. And to show how disorganized I am, I couldn't remember the title of the talk. I apologize. Tim will talk to us about AI in a world of increased access to societal data, but from within TRE environments, or TREs. Tim, over to you, I think.
It's only water up here because I've got a terrible sore throat and I'm likely to pass out before.
Behind you.
Behind you? Okay, this is like a Sanger pantomime. Okay, that's the title I put in. I'm actually going to talk slightly more broadly than that. So these are the things I'm going to cover. What can AI do for infrastructures? Because, of course, Director of ELIXIR, that's all about infrastructures. But then what's infrastructures going to do for you if you want to train AIs, which is more what the talk was about. So just a little bit of introduction of what ELIXIR is, just in case people don't know. It's a sustainable infrastructure for biological data across Europe. It's got 21 country member states, and then it's got EBI as a member as well, as a non-country member. And it's organized as a set of nodes. So every country that's a member has got a node coordinating activities in that country, frequently with national funding, many times with significant resources. Obviously, the heavy lifting, a lot of it's done by the EBI. They host most of the large-scale infrastructures, but there's a lot that's distributed as well. And the hub, we don't hold any data. we just coordinate across this network. And the structure covers compute, data, interoperability, tools, training, facilitated through a whole load of communities of interest groups across this network. So this network is 21 countries, about 250 institutes, about 1,000 people directly involved. We had our all hands in Uppsala a few months ago with 500 people there. So it's 10 years old now. It's quite an established structure for coordinating bioinformatics, resources, training, etc. And if you want to read more, then there is a lot online. There's documents on the current plan of 5 years, 24, which has just started to 28. So that's all I'm going to say in the sort of background bit. Okay, so the function of infrastructure, as far as I'm concerned, it's to enable research. We're not doing research, we're trying to enable others to do research. To enable it, we're trying to lower transaction costs, just make it easier for you to use the data that's available. And I think back to the days of the Human Genome Project 25 years ago, where suddenly we had all this genome data for human, and it was incredibly difficult to use. And Ensemble came out of that concept that we needed to organize that data in a structured way so that people could then build stuff on top of it. So a lot of this is around standardization to allow interoperability, reuse of tools and resources, and then also pre-calculated data that researchers can build on. So where can AI help in this? So various things that ELIXIR is involved in. There's a machine learning focus group. It came out with the DOME recommendations around definitions for machine learning. And there are model repositories under construction using some of these standards. So EMBL has the biomodels repository, Euro Bioimaging, which is another S3 infrastructure. ELIXIR is one of the S3 infrastructures. That's a European Strategic Forum of Research Infrastructures. There's a group of biological infrastructures that work together. In fact, we just had a strategic meeting yesterday in Brussels, which I've just come back from. So Eurobioimaging is building bioimage models, for example. The other thing is where there's pre-calculated data that communities can use. The most obvious example of that in the AI space, of course, is AlphaFold. You have this algorithm, you can run it to predict a structure. But getting it set up to run on your favorite sequence, it's a reasonable amount of hassle. It's a lot easier if somebody's done the pre-calculation for you, and that's what EMBL did. We're building this database of predictions now, hundreds of millions of predictions there. And that significantly changes what people can do. It's already there. I can just go and look at it, explore it, look at relationships between them. I haven't actually got to do the building. I haven't got to work out what's the best way of doing prediction, for example. Then you've got the opposite side of AI, LLMs, chatbots being integrated into various service interactions. Again, about making it easier to access things. And we're just at the start, I think, of that being played around with by different resources everywhere. There's significant potential with literature. There's already, Europe PubMed Central is run by EMBL here. It's one of the ELIXIR core data resources. It's had NLM functions for a while, but there's a lot more, I think, that you can do as a community in AI in this space. So there's a mess in the literature. This is quite old now, The Economist railing against non-reproducibility of science. And it's significant. The Economist writes about this. It's on the front page. It's an editorial, not to mention an article. Policymakers take note of this sort of stuff. This was saying basically only a tiny fraction, around 15% of publications pointing companies in a direction of a new entity or direction were reproducible. There's a huge lack of reproducibility. Can we use AI to clean that up, to help flag things that are inconsistent? So people go and randomly search, see a journal article, and if it's already been flagged as not being useful, maybe that can be done in advance. There's also a lot that you can do beyond that. So my friend John Molt, of course, of CASP fame, is trying now to go beyond predicting structures, the CASP competition, to can you interpret different mechanisms for function. So here's Alzheimer's, ApoE4. These are all the different mechanisms that have been proposed, or a lot of them. And the question, they can't all be right. Which one's got the strongest evidence? Can you do anything about that without reading hundreds of, probably thousands of papers? And can you synthesizing something out of that? And so he's been using ChatGPT for that. And this is his sort of summary of what he thinks you can get out of using those sorts of tools to interrogate the literature. ChatGPT's read all the literature, all that's accessible, and you can ask it sensible questions about, with a certain amount of prompt engineering. In fact, we were together on holiday and we spent the early mornings torturing ChatGPT about a year ago with this, but he's now done this in a more systematic way. So I think there's a lot that you can do if you have this pre-calculated, in this sense, LLMs that have read everything. You've got a uniform way, to some extent, of querying the literature, trying to query what's in the literature, rather than actually having to go and read it. So there's a lot we could do in this space, but there is an aspect of need for political lobbying here, because, of course, even today, only half the literature is open access and can be read by these sort of open algorithms, that are reading these things. I think there's a good argument now to go back to the policy makers and say, look, before the argument was, you're rich countries, you can pay for the subscriptions to these journal articles. But now, because we could potentially leverage on top of this by putting them through an AI, if that's blocked by the publishers, that's a huge disadvantage for the whole of global research. This is a good example of where you institute a compulsory license. That was done in various other critical spaces before World War I. They introduced a compulsory license for patents around aircraft manufacture because it turned out they're actually quite important when you have a war. So I think there's an argument for pushing to try and open up some of these spaces. Okay, so that's the first bit, infrastructure. Where can you usefully apply AI, or some ideas at least, which you might be thinking about in these sort of communities you've been talking about. Right, what about the other side of things? So I look at the whole of biology as being a grand project. We're trying to understand biological systems. And of course, 25 years ago, we started to have whole genomes. And of course, the problem was interpretation. I'm afraid this isn't a very EDI compliant cartoon. But it was 20 years ago. And having got the whole genome, the hope was, of course, that you could predict everything, all the way up to the existence of rice pudding and income tax. Of course, the reality is our prediction abilities are pretty limited. And what we've actually had to do is collect a lot of other data at different levels and organize that. And then, a lot of the resources that are the EMBL, scattered around the world, across or coordinated by ELIXIR, are these resources to try and give you these intermediate components and hopefully organize them. But ideally, this is an opportunity for AI. And of course, one of these components, AlphaFold, has made an impact there, a major impact in terms of this being able to predict all the way up to whole organism systems and even ecosystems. And I'll just say on the AlphaFold, there's various sort of discussions that go on saying, well, it's just managed to organize the fragments of structures we already know. But I will point out this paper, which is also John Moulton Osnat, deep analysis of the actual predictions saying, no, it's doing better than that. It really is, it's essentially models something like the physics potential that we have been unable to model in molecular dynamics. It produces very accurate atomic level coordinates, configurations of atoms which we have not seen previously in the database. So that's just an aside. Okay, go back to this. So we have AlphaFold making this step from this collection of protein structures. What about using AI in other types of ways against these sets of data? Well, probably we need more data. And so this is the point about TREs, additional sources of data. Up to now, we've lived in this research space researchers get money from grants, go and collect some data, organize it, but it's a limited set of data. Of course, the advantage is it's frequently open, although that's not always true when it comes to cohorts of human data. But the point is that there are new sources of data because there are certain classes which are routinely collected. And obviously I'm thinking of health, but there's lots of other areas as well, in the environment, surveillance, etc., social science data, government data, that could potentially be analyzed if we had access. And the problem, of course, is restricted access. So that's a huge opportunity for massively expanding the sorts of data that's accessible potentially for AI, for training to try and deeply understand relationships. Now, of course, that routine collected data is frequently noisy, so that does make it harder, but AI is supposed to be good at that. And then, of course, it's restricted, which is, of course, the biggest challenge. So in this space, this would mean adding in clinical genome sequences, at least as one starting point, and the clinical medical records. Of course, we have been doing this in the UK, but the European Union has got a very clear picture of how to do this as well. And this is called the European Data Spaces. And this is not just restricted to medicine. There's 15 planned data spaces right now. There was a meeting with the Commission yesterday about this. In the health space specifically, they're planning the European Health Data Space. There is a regulation that has gone through the European Parliament to enable that creation. Basically, it's going to require that health systems across all EU member states are accessible for research analysis. Then, of course, there's parallel projects. That's the electronic records. But of course, there's huge data sets in genomics, and there's data sets in imaging, which potentially can complement that. And those are in this list of infrastructures that collectively can give you a very large data set of data. Of course, when you think about human, how far have we got in the UK? We've 500,000 in Biobank, 200,000 with Genomics England, 1 million is planned in Europe, but if you're talking about records across Europe, it's 430 million, everybody. So the other thing is that if you build this infrastructure correctly, you've got the potential to do something else as researchers. Everybody's worried about impact. That's what the funders start asking questions about. The old way of presenting this is, well, we'll have a drug in 20 years. But there's new alternatives, which is if I've got the data and it's actually real-world data, patients now, you can potentially build algorithms, test them on the research side, and then deploy them on the clinical side, maybe in the same infrastructure, and use them for clinical care if you can show effectiveness. So there's an opportunity to apply digital analysis of data if that data is already being made available for research and it's close to active use. And remember the opportunity in terms of the scale of money in the health system compared with the scale of money available for research grants. Of course, the problem is privacy. So, if you can apply that other side, then essentially it means now not just having the genome sequences, but thinking about the variants, doing prediction all the way up in terms of clinical effects, and being able to feed those sort of things back. And of course, that's what Genomics England's been doing in terms of rare disease interpretation, having a pipeline that runs in parallel for clinical care. But Europe is building this. There's the 1 Million Genomes Initiative, and this has been implemented, and a lot of this is coordinated by ELIXIR. So ELIXIR coordinates the B1MG project, which is the kind of the organizational side of working out what the policies are, the implementation. But more significantly, it's been coordinating the GDI project, which is to build a federated genome data infrastructure across Europe so that you can hold genomes securely in different member states, but also federate between them. And this will end at the end of 2026. And that's linked to all the other bits that are being built, the components that are happening purely in the health system, like the European Health Data Space. The idea that the sort of subset of genomes, the infrastructure will be built for initially researched genomes, but progressed, but able to support clinical genomes as they start being collected. And there are countries that are doing that. Denmark, for example, has about 20,000 whole genomes, very similar to the UK, collected for clinical care, but enabled for research. So in terms of infrastructure, what this means is the TRE model. Up to now, the starting point for GDI is a set of research volunteers going into a data center, being made accessible for research. The next stage is you start getting clinical genomes into that infrastructure. But once you've got that far, you might as well build the other side as well, support the idea that you have apps that have been trained on the research side and deploy them on the research side. Now, this, of course, is the model for TREs. This seems to be a TRE-enabled audience, so maybe I don't need to go into this very much. But obviously, it's the 5 safes that you have to implement. Safe data, that the research side can only see properly de-identified data. Safe setting, where you don't distribute the data at all. Safe people, and safe projects to make sure everything is the kind of analysis that's supported by the community with transparency. And then most important, safe outputs, which is where the title came from. So you're only allowed to export stuff which is not identifiable. And that's where you get a problem when you start thinking about AI. So the potential of all this is massively large sources of data, additional outputs in terms of impact of your work, all enabled by doing infrastructure design in the right way, so it can face both ways. But it's not just health. There's all kinds of societal challenges which involve biological data. They all rely on data and science. And so the data spaces are, again, not in just this space. There's the Green Deal, which also has a data space. So the whole sort of biodiversity. In fact, now we're talking about One Health, about how all these things interact: human health, pandemic, biodiversity, food security. These are all interconnected issues now that, with climate change progressing at the speed it is, are suddenly very relevant to think about. And, of course, an example of what has been built is what was done for COVID, where there were data hubs that were set up for the data collection. EMBL built a database for this. ELIXIR was involved in standardizing the metadata because it coordinated by COVID, the European project. And we've ended up then with tools sitting on top of that, Galaxy, which is part of the ELIXIR group, and a toolkit for doing this. And of course, now we're talking about how can this be sustained as an infrastructure. It's societal data. It's data that's being collected partly as part of public health systems worldwide. But in terms of AI outputs from all this, the biggest problem then is how do you get this stuff out? And there is this DARE project in the UK, which is essentially UKRI-funded initiatives of trying to work out all the missing components of if you want to do trusted research environments and the different impacts of that. And one of these projects was Grail Matter, which was looking at what happens if you train an AI. And so I think the summary of this is, if you've got a TRE and you've trained an AI inside, you've got to worry about the airlock process, getting stuff out, which has got to be compliant with safe outputs. And the problem there is, how do you assess if your AI model has not captured some private information? There's quite a lot of analysis of that, and people investigating whether they can prove that the AI doesn't capture it. But up to now, it looks like you can't be certain of anything. And in many cases, even when you think it's safe, it turns out not to be safe when you can probe it in various ways. So if you can't check AIs for privacy, can't be confident, they're going to need to stay in the TRE, which means that if you're a commercial group that has trained something, because one of the advantages of TREs is that because you're not distributing the data, it's much easier to have academics working in there, commercials working in there. But if you can't take the AI out, what do you do? maybe the TREs could offer a service. You could run the AI you've trained inside that, but that requires a service wrapper around it. That way you could at least prevent the AI escaping with all the data that is captured. That's one of the things that's proposed by this DARE-funded project. And then that's bad enough, but what about when you're in this federated landscape? European Health Data Space, the model that's being proposed is a set of federated, what they're calling secure processing environments. And in fact, in the UK, with the health system here, we're building 11 secure data environments for NHS data. The idea is you want to do training, investigation over a large data set, you're going to have to federate. And if you do federated learning, what do you do in terms of the security? Can you set up an enclave that's holding the AI like a sort of super TRE that can connect privately to the other TREs? The main problem with the main reason for having federation is that people don't want the data to move. And certainly in the national context like the European Union, you're talking about getting countries' healthcare system data to benefit the EU by analysing across that. But I can't see any member states, I haven't done enough engagement with the public anyway, I can't see member states being prepared to move blocks of data en masse. Whether they'll consent to federated AI training when there's worries about exactly how much data is then flowing between systems which capture the AI is a different question altogether. So these are the sort of issues. They're all part of this general problem of data management in the life science with the scale we're at now and the opportunities we're at here. It's not just data growth anymore, it's the diversity of data, where this data is located, because in many ways ELIXIR was set up when a lot of the data could come to EBI. But once you're talking about data sensitivity, it's not going to move around like that. If it's health data, it's going to stay probably in the countries, and we're having to build systems that support that. There is an ongoing EU-funded project, again coordinated by ELIXIR, called EOSC Entrust, which is addressing, it's working on many of these issues around how do you do federation securely. Okay, so I'm going to stop there. Acknowledge the people in the hub and the nodes, Embal. Carol gave me one of those slides, and of course my history in Genomics England, King's, Health Data Research UK and the NHS over the last 10 years. Thanks so much.
Are there any questions? Unfortunately, I've put the iPad down, so I haven't got Slido.
I was wondering, do you see more interpretable AI models as almost a solution to the problem of having AI services within trusted?
Well, you mean in the sense that they might be safer?
Yeah,
Ronnie.
Are you meant to be catching a...
Yeah, it's going to be lethal, but...
Oh, I see, that's a microphone.
Can you hear me?
Yeah, I can hear you, yeah.
Sorry.
My question
I
was
mean, I think what did you say? Not interpretable.
so just if models are more interpretable and
Yeah.
explainable.
So, I mean, I think that will help from the point of view of other things. The type of stuff that John Malt's doing. If you want to try and investigate which of these 30-something mechanistic explanations, then you'd really like some interpretability where it says, I don't think I agree with this model because, which isn't entirely clear that they do that right now. So that will help, but I'm not sure it will help with the TRE problem that much.
Sorry, Martin, I don't think we can get a microphone up. No, we can give it a go. While we take microphone, I've got a quick question for you.
Yep.
NIH mandated open access. We've had green, we've had gold.
Yes, well, you see.
Why, do you think? I mean, we know why.
So look, there's these two worlds. There's the open access world, research data, which is not sensitive. We've been sort of progressing in the direction of actually, when it comes to personal individual genomes, it's not even your genome, it's your family's genome. All your past and all your future children to some extent. And it's sensitive, and we don't know how sensitive yet. because, what doesn't seem sensitive right now may be more sensitive in the future when we get better at interpreting genomes. So people are very nervous about this. Certainly at the moment, I think it's acceptable that you don't distribute this data. So, to some extent, I'm hated by both sides.
Okay.
Sort of the open people think I've reneged on it, but frankly, the other side, you have to respect that privacy is a really serious issue. If you want, particularly, not just to have a few volunteers, all the volunteer cohorts are biased in some way. If you want to have whole health systems, and then deal with all the different things that are hidden in there in terms of inequitable treatment, biases, etc., you really want to encourage people that we're going to do this in a secure way. And that means building infrastructure. And it's inconvenient for researchers as a starting point using these things, but of course we are getting better at building them now. And there's been a lot of publications coming out of Genomics England. People complain about that was one of the first large-scale TREs, but it is usable. You can get stuff done.
Yeah.
I have a postdoc working on this right now.
Martin.
How do you think we're going to deal with the problem of bringing the data to the hardware? Because a lot of these TREs don't have the computational resources to train AI, and a lot of, for example, if I want to do a very large model training right now, I need to send it to somewhere like Baskerville, but that's not a TRE. And every person who owns data wants it to be in their TRE, not someone else's TRE. So you sort of end up with a situation where you can't train it because the TRE doesn't have the resources.
So I think you have to bite the bullet partly. There's a federated training system across London hospitals. They've actually installed some GPUs in every single hospital. And actually that gives you two advantages, because not only do you have to not move these big image files, but you then have a GPU system that you can run models on once you've trained them and deploy them in the hospital. So there are federated structures like that. Yes, you've got to buy some hardware. Genomics England, in the sort of the first iteration of the infrastructure, it's standard HPC type structure. But the second one that was used, built mainly for COVID initially, was to wrap AWS and do all the security structuring so that it looks the same, but you can, through LifeBit, you can deploy stuff inside AWS. So in that sense, the data's moving, but it's inside our security envelope. And the critical thing there, I think, in terms of public confidence, is customer-managed keys. You must have the agreement. I mean, Amazon actually, they've got a lot of security documents on this. They want to be able to say, we couldn't look at your data even if we wanted to. They will need that if they're going to sell services to banks. So the big cloud providers actually will do a lot of the security engineering and basically give you key management. And I think if you have key management, then you can say it's your TRE. And that's a sort of one viable route. But yeah, this is always going to be, you're getting access to a vast extra collection of data that you've never had before. There's a price for that, which is you have to live with the structures that the public particularly find acceptable.
Unless there are any more burning... Okay, one quick burning question. Rob.
Yeah, so if you can't link the data between the TRAs, would it be more acceptable to have a semi-trained model which you basically move around between TRAs?
So then, so this is the question of, it's partly about TREs and trust between TREs. And the NHS is sort of, if we can make an accreditation framework, you have to jump through, it's like ISO but worse, which in fact does exist in the UK for social science data, and is now being attempted to tweak it to deal with health data. If you can then say, well, all these TREs accept this, then maybe it's easier to move data between them or to move models between them. Because then all the exits are the same. They've got the same level of rigor, all the airlocks, so there's no way of moving it over here and then there's an easier airlock route out. You've got to basically make sure the barriers are the same all the way. It's just like the European Union building a common frontier.
I'm afraid we're going to have to call it time. I apologise, people on Slido. I didn't pick up the iPad. Any other questions? Sure, they come to Tim. I'd now like to introduce Mafalda, who comes from CRG in Barcelona. For those who work at GenGen, they probably know it well. Mafalda's background, a lot of Bayesian learning and generative models. Particularly predicts the effects of genome variation on phenotype. So obviously this has got direct impact on the diagnostic yield of patient sequencing. Mafalda's talk today is on, I can read it from here too, models with Jesus, my English is terrible. Models with interpretable scores for discovering pathogenic variants.
Well, thank you very much.
Over to you. Sorry.
Thank you very much for having me here. I'm super happy to talk about my work. Here. Yeah, so I think we're going to dive a little bit more into sort of the research enabled by the kind of infrastructures that Tim was talking about. And what I would like to talk to you about today is an unmet need in clinical genetics of trying to pinpoint pathogenic variants in patients. So as sequencing is becoming available for diagnostic care and preventative care, we have this necessity of identifying what could be the causal pathogenic variants in a patient. And today I'm going to be talking precisely about identifying pathogenic missense mutations, and this is really a needle in a haystack kind of problem, because as an average person has about 10,000 missense variants in the genome, to try to identify what could be the pathogenic variants is kind of like a needle in a haystack problem. There's been a lot of development over the last decades in statistical genetics to try to collect bigger and bigger cohorts of patients and across the population, and develop tools in GWAS and protein analysis, but these methods still remain limited, especially in the long tail of rare variation. Often there's sort of gene-level statements rather than variant-level statements. And this is a problem for pathogenic and oligogenic disease, but it's also a problem for monogenic Mendelian disorders, where you would have imagined that it's where it's easiest to identify what is a causal variant, because it's kind of like a large effect variant that is driving the disease. But actually, the vast majority of patients that suffer from Mendelian rare diseases still don't have any kind of genetic diagnostics. And part of the problem is that being rare, these diseases are just harder to study. It's harder to put the cohorts together, and it's also, even when we have large cohorts, they're limited in scope because it's quite hard to identify what is driving diseases that are as rare as one. So one approach that has been very fruitful in the last decade is to kind of decrease drastically this haystack by just looking at de novo variants, so sequence trios, and by that I just mean sequence the parents of the patient to try to identify the de novo variants. And this is powerful because it has been shown that severe disorders, rare disorders, are often driven by de novo variants. And this is kind of, here in this plot, you just have an example of a cohort that was actually put together by people here at Sanger from the Matt Health Group of 31,000 trios of patients with developmental disorders. And you can see that there is a large excess of de novo missense variants in this cohort, kind of like pointing towards the idea that these patients have causal missense de novo variants. But the reality is that even if this looking at de novo variants kind of reduces the problem and make it easier, most of these excess missense variants are still unexplainable and outside of genes that we know are involved in developmental disorders. And also trio sequencing is also difficult to put together. It's expensive and it involves the participation of both parents, which in many cases, in many settings, it's quite hard to obtain. But the flip side of this is that it points towards quite a simple genetic architecture of these disorders, and really is just saying that if we just are able to find that causal variant, we might be able to diagnose a lot of patients. So this seems like a great setting for computational biology and for deep learning to try to address. So the idea is that we have what we would need, I guess, to develop a model that is able to identify or pinpoint causal mutations, pathogenic variants in diseases as rare as one, and perhaps even across the whole exome rather than just looking at de novo variants. We just need a model that has a meaningful notion of severity, kind of an interpretable notion of severity, such that I can identify that there are some variants that are pathogenic or might be involved in some disorders, but there are some that are really outliers and might be driving extremely pathogenic and extremely severe disease. And for this, we need tools that satisfy two conditions. First of all is that, like I was just saying, this pathogenicity score really meaningfully reflects variant severity. And also we need this score to kind of hold for the whole exome, because we want to study variants across the whole exome of these patients. And I'm going to try to make these more concrete. So some years ago, together with collaborators, we were exploring the idea of looking at variation across diverse species as a way to identify pathogenicity in variants in humans. And the idea is super simple. So if we just look here, it's protein sequences, or it's supposed to depict protein sequences across a protein family, across diverse organisms. The idea is if we have a model that is sufficiently expressive, that can capture patterns that are being preserved across millions of years of evolution, preserved, co-evolving together, then we are able to capture constraints in sequence space that might be related really to function of this protein family. And the idea is that once we have such a model that describes the distribution of these sequences, we can then ask if a new sequence with a variant that maybe we never saw before in a human, how likely it is according to this model. And the idea is that if this sequence is an outlier according to this model, maybe this variant is disrupting some important pattern of constraint in sequence space, and it's probably associated with lack of fitness in this protein and probably associated with disease. So this is kind of like a super simple idea, and but actually it works pretty well. So we for this specific work, we used a variational autoencoder, but there's different tools now that use different kind of deep learning models with kind of same idea and ethos in mind. And so just to give you an idea of the kind of performance of these models, so we call this model EVE, and this is an example for P53 and P10, and here magenta are clean VAR known pathogenic variants in these genes, and the blue or teal are known benign variants in these genes, and you can see that the model quite separates very nicely benign from pathogenic variants. And not just that, but it has similar performance to deep mutational scan experiments, so experimental assays to try to address this exact same question. And we did this at scale to every single gene that we knew was involved in disease. And okay, for some genes we have more data for validation than others, but across the board we see that there's quite good, or these models have quite good power at separating benign from pathogenic variants. And across the last few years, there's been a lot of development in this kind of space. There's a lot of tools that kind of address this question, and they are actually quite excellent at separating benign from pathogenic and predicting clinical data like I was just showing you. Now the problem is that the interpretation of these models is not very clear. And when let me just give you an example. When we look at the distribution of scores for genes that are aptly insufficient, so definitely involved in severe disorder or involved in dominant disorder, versus loss of function tolerant genes that are effectively non-essential, what we see is these models have very similar scores for both of these kind of families of genes, where you would expect that you would see a much stronger tail of pathogenic variants in genes that are involved in disease compared to genes that are non-essential. In the end, what is happening is that these models are excellent at ranking variants within genes, but they really fail. So they're very good at predicting if a variant is going to disrupt molecular function in a particular protein family, but they fail to capture how impactful that gene is for human disease, and therefore how this relative severity across genes really impacts human health. Another way to look at this is that they kind of, all of these models provide a continuous score, but the interpretation of this score drastically changes from model to model. Instead of having a score that maps to disease severity, what most models are providing is a score that actually reflects a probability of a variant being labeled as pathogenic or benign. So many models have a bimodal distribution. Just here is an example of alpha-missense, where close to 0 are all the benign variants and close to 1 are all the pathogenic variants. And instead of this score being reflective of severity, it just basically says, the model believes that this variant might be involved in some disease at some point. So with this kind of model, it's really impossible to identify severe pathogenic variants, as you would have, all the variants mapped to a score of, I believe this is a pathogenic variant. In a sense, you can already see that this distribution of scores cannot really be a distribution of severity, because it's bimodal, which goes completely against your expectation of negative selection on severe pathogenic variants. So to try to gain a notion of interpretation in these kind of models, we opted to take a strategy, which I believe is a strategy that we're going to hear over in this conference, which is to build a simple model, a model where we can have explicit model assumptions on top of a highly expressive model, that's a deep learning model that really is not very explainable and not very interpretable. And to do this, in this particular setting, we leverage variation on 2 different timescales, so variation across the tree of life, like I just described, that just models constraints across evolution and constraints in protein families and protein molecular function, and also model the variation across a human population, so model human-specific constraint that hopefully maps to human health. The way we do this is to first compute these variational anticoders, large language models across diverse organisms for every single protein in the human proteome. So we have about 20,000 of these models, and for this we get an evolutionary score like the one I just described that has very good variant level resolution at distinguishing variants that are impacting protein function and molecular function, but are not very good at distinguishing if these variants are important for human health. And then on top, we change the problem and rephrase it as the capacity for the model to predict if a variant is seen or not in the UK Biobank. So we just use this as a very simple model. A model, well, it's just like a Bernoulli process with a Gaussian process. In fact, if that function f was just a linear function, this would just be logistic regression, but we allowed it to deviate a little bit from linearity. I'm happy to talk about this later if you're interested. But the key aspect here is that by doing this, because we have variation across the whole population, across the whole proteome, by predicting if a variant is seen or not in the UK Biobank allows us to have some sort of information sharing mechanism across all of these protein family independent evolutionary scores. And what we get is that this model rescales the scores for each protein family according to how important it is for human health. The result then is, bringing back the example I showed you earlier, is that we see that for haploinsufficient genes, now we really see here, we call this model POPI just because it has population data. We can see that more negative values are more pathogenic, and we see here a tail of negative variants in genes that are involved in disease compared to genes that are effectively non-essential. So the kind of behavior that we would expect from this kind of model. And when we look at the distribution, so this is the distribution of scores across all the variants in the UK Biobank, in opposition to this kind of bimodal distribution from models that are just trying to predict if a variant is benign or pathogenic within a protein. What we have now is kind of a bell-shaped curve where you can see that it is negative tail corresponds to the more pathogenic variants across the population. A little bit more like we would expect according to negative selection on these variants. So what can we do when we have a model like this and we apply it to try to identify pathogenic variants in disease cohorts? So we looked at, again, these 31,000 trio cohort put together by the group of Matt Hurls, and we see that the model is quite good at distinguishing cases from controls. So these are the de novo variants at the de novo variant level. And what we see is that at the benign end of the score, so around -4, -3, we cannot distinguish if the variants belong to a patient or a control. But what is very interesting is that for scores that are below here around -5, we have, yeah, exactly, we have about 99% confidence or recall that these patients, these de novo variants are really variants in a patient rather than a control. And you can see that this corresponds to almost like a 10-fold enrichment in variants that are deleterious at these scores. So we can use this to try to identify variants that are potentially causal. And when we do this and see in which genes they are, we recover a lot of the genes that have been found in this study before. We also find a lot of genes that have never been identified in this cohort but are known to be involved in developmental disorders. And we also find evidence for genes that were not known to be involved in developmental disorders before. And actually, since we put this paper in the bioarchives, 20 of these 120 genes have since been identified in familial studies, have been identified being involved in the developmental disorders. So really sort of giving us some confidence that maybe the kind of evidence that we are bringing is meaningful. And I was talking about showing you results of how to identify pinpoint de novo variants that are causal, but then we can try to do this exercise in trying to identify or pinpoint variants that are causal across a whole exome. So it's a much harder problem. But we again see that in the lower end, more negative end of the scores, we can start to identify patients and variants that are associated in patients. And in fact, we find that in 98% of the cases that we identified with de novo analysis, just by looking at the exome, we recover them and we identify the variants, the de novo variants, as being the most severely pathogenic across all of the variants, included the inherited variants. So giving us kind of a feeling that we can use these kind of tools without trios and really just directly at the exomes of the patients. And this kind of gives the same kind of message, this plot on the right. This is just the percentage of the cases that are putative diagnoses, so people that have variants in genes that are known to be involved in developmental disorders, and with a de novo variant there. And just by looking at their whole exome, we can see that we recover a lot of these, a lot of the variants and causal variants without the need for trio data. So just to summarize, until now, methods did not provide, or methods, variant effect prediction methods, did not really provide an interpretable severity score, and instead provide a score with a difficult to parse, a continuous score that is difficult to parse and with a difficult interpretation and mapping to severity in humans. And by leveraging variation on different time scales across the whole tree of life and across a human population, we managed to build a model that sort of places variants in a proteome-wide spectrum of severity. And this can help us to identify variants in disorders that are as rare as one and potentially without the need for large cohorts or trio data. But I would like to finish with a comment on where we are going next and what kind of things are we thinking about next. Certainly, severity is not one-dimensional, and what interpretable models mean can change very much. Certainly, there's 3 topics that are very important. I didn't talk about inheritance patterns, and the model that I've just described has some difficulty in distinguishing if a variant is mildly pathogenic but associated with dominant disorder or severely pathogenic in a disorder that is recessive. This is something that the model has some troubles dealing with it, and this is definitely an axis that we're interested in exploring. Variable penetrance is also another axis in this severity, non-one-dimensionality, and certainly is something very interesting, and again, pointing to what Tim was talking about. there will be different mechanisms or molecular mechanisms driving disease and driving this pathogenicity, and it would be great to develop methods that can disentangle all of these different mechanisms. And all of these areas are areas that I believe really would benefit from models with explicit model assumptions, so that you can find more interpretable solutions for these predictions. And this is something that we are currently working on in our group. And this is CRG in Barcelona. This was work that was done in my group at CRG and also at the group of Deborah Marks at Harvard. In particular, Rose, she's a PhD student that worked very closely with this work. So thank you very much.
Hi, do we have any questions? Just in case there are any.
Yes. I don't know if you will be given a microphone. Yeah.
It's for the people online.
So one of the standard ways to create interpretable scores in machine learning is to calibrate the scores using techniques like isotonic regression. How is your method different?
Sorry, can you repeat your question? I'm not sure if I heard it.
So often people calibrate scores in order to make them interpretable using methods like isotonic regression. And so I was just wondering how different your method is from those.
So this method really just uses the presence of or not of a variant in the UK biobank in the human population. So really it's reminiscent to methods to assess the how the essentiality of genes, so methods like SHET or PILI, where you're just basically looking at the amount of loss of function variants across the population in a gene. These methods are very powerful, but they're really only gene level statements. And here we're just trying to use something that has the same sort of philosophy, but that will allow us to have variant level resolution. But that's kind of the calibration, quote unquote, is just really leveraging variation in the human population.
Unfortunately, we're out of time, so I'll have to call an end to questions. By the way, that's a beautiful picture. And thank you very much, Mafalda.
Thank you.
Next up, we have Miha, who unfortunately I didn't have time to talk to. So I'm just going to have to read this out verbatim. Miha is the founder and CTO of Genialis, the RNA biomarker company. He's led the development of a pioneering biomarker based on RNA-seq and machine learning, which was accepted by the FDA for clinical trial use. He's passionate about data integration and XAI. Miha has also co-developed the Orange data mining suite and Resolvi bioinformatics engine. He earned his doctorate in computer science at the University of Ljubljana and completed postdoctoral research at Baylor College of Medicine. His talk is on explainable AI and precision oncology and how the, Zerna TME panel, an ML-based transcriptomic biomarker, earned the confidence of drug developers and regulators. And I apologize for murdering that.
Thank you. So I'm an AI geek by heart, but I'm also someone who cares deeply about giving back to the community. I was a scout leader, I contribute to open source and open science, and my work at Genialis is really all about one thing, which is helping patients get to the best outcomes. I have the privilege of working with some talented individuals. We have 14 PhDs on our team, two of them are MD PhDs with deep expertise in healthcare and machine learning. We are also a founding member of Alliance of Artificial Intelligence in Healthcare, which is an advocacy group that promotes responsible use of AI in medicine. So I'm happy to invite you to the panel in the afternoon when we'll talk about that. To set the stage, biomarkers, the way I define them, or we define them, are measurable indicators of a biological state or condition, and they're particularly useful for precision medicine because they can tell us about the disease phenotype from which we can guide treatment decisions. You have different kinds of biomarkers. Examples here would include you go to a doctor, you're given a prognosis, right, based on the prognostic marker. What I'm going to talk about today are predictive biomarkers. Here's where we focus as a company, so that we predict patient response and patient selection for clinical trials. Our biomarkers process RNA data. That's why we call ourselves the RNA biomarker company. Standard of care today, particularly in cancer treatment, relies on single analyte biomarkers like the presence or absence of a mutation or gene. We, in contrast, develop complex markers of human biology that better define the phenotypes of disease, and then we link these phenotypes to specific therapies. Here we have two examples. To the left, for KRAS inhibitor treatment, the treatment is assigned based on the presence of G12C mutation in the KRAS gene. What we did is we modeled the pathway upstream and downstream to better capture the relevant biology. To the right, you see our model of tumor microenvironment that captures the interaction between angiogenesis and immune response. And I'm going to talk about both of these models that are built on RNA-seq and machine learning in two of the use cases. We model cancer biology through hallmark signatures. By signatures here, I mean predictors that capture some key aspect of tumor behavior. These signatures, in our case, would use RNA, which tells us about the tumor functional state at a particular point in time. My team has built about 100 validated signatures that we'll have a closer look. The methodology that we use to develop these RNA biomarkers for patient certification and selection is called Responder ID. We've built a foundation model of cancer biology that integrates the signatures that I mentioned before, and we can use this foundation model to map RNA-seq data into disease biology state, where we can then use background domain knowledge, like in this illustration, biology-informed filters, to infer the relevant phenotypes. I'll briefly explain the foundation model, and then we'll have a look at the use cases. Just simply put, right, what does it do? Our foundation model organizes cancer patients into clinically meaningful subgroups. We've heard a lot about large language models, LLMs. The foundation model that we've built is a large molecular model, LMM. So what it does is it maps RNA-seq molecular data into cancer biology space. So specifically, each patient's RNA-seq sample is characterized with about 100 scores, where each of the score corresponds to a particular biology signature. Let's take a closer look. We integrate specialized machine learning models where each of these machine learning models captures unique biology. I'll showcase the tumor microenvironment model in the following use case. A key component here is our universal space transformer. That's a deep neural network that adjusts the bias and adjusts the variance of RNA-seq data. And the output of our foundation model are 128 signature scores that I mentioned. Now, the second part of Responder ID methodology is that we fine-tune this model to patient outcomes. We infer disease phenotypes based on biology, and then we link these disease phenotypes to therapies to make informed treatment decisions. The foundation model was trained on both public and proprietary data, we ensured global diversity by working with data providers or cancer centers such as Sidra Health in the Middle East, Academia Sinica in East Asia, 4Base Care and Cepien Biosciences in South Asia, and in the North America we work with Tempus, the Moffitt Cancer Center and Exact Sciences. How we put this foundation model into practice. In the first example, I'll talk about Zerna TME panel, which was developed in collaboration with Onccerna Therapeutics. The biomarker panel is now in prospective validation in clinical trials. The panel identifies dominant biology in tumor microenvironment. It predicts 4 subtypes that are characterized here as angiogenesis, immune suppressed, immune active, and immune depleted. You can also consider it on 2 axes, 2 phenotype axes, based on this immune score and angio score. So both angiogenesis and immune suppressed phenotype would be high in abnormal blood vessels expression, right? And immune depleted and immune active would be low in these blood vessel expressions. The same goes for the immune axis. The TME panel workflow starts with processing tissue samples, right, and extracting RNA, and ends with this discrete phenotype prediction illustrated here in one of the 4 subtypes. We work typically with FFPE tissue samples from recession or biopsy. The RNA is extracted and sequenced, and then analyzed by this universal space transformer, followed by a shallow neural network with 1 hidden layer and 2 neurons inside. We chose a simple architecture on purpose because it allows us to unbox it, as you'll see in the following example. The plot to the right is a representation of this latent space of the 2 hidden neurons that actually capture the 2 relevant biologies of the tumor microenvironment. We looked at the weights in this neural network. The genes that we selected were curated from the literature. Illustrated to the left is the immune phenotype, and we selected about 100 genes, some of which were related to the immune phenotype, others were related to angio phenotype. When you look at the model weights of the 2 neurons, you can see that for the first neuron, it assigns the neural network inferred that on its own, right? It assigned high weights to the immune genes. And the second neuron was assigned high weights to the angio genes. But you can also see some interplay between the 2 biologies, because some of the genes are relevant for both neurons. If we look about the 4 phenotypes, just to see how it matches our hypothesis, so we did gene set enrichment analysis to MSIG-DB signatures. The signatures were not, they did not overlap with our gene signature. And as it turns out, angio phenotype and immune suppressed phenotype is related or has high activation scores of angiogenesis and mesenchymal biology signatures. And in another example, when you look at the immune high phenotypes, which are immune active and immune suppressed, they have high activation scores for the inflammatory response signatures. You can also see that the immune suppressed phenotype, in one case, it's related to the angiogenic phenotype. In another case, right, it's also immune high, this immune suppressed, it's related to immune active phenotype. But really, when we explored it a bit further, we could confirm that this is indeed another separate, distinct phenotype from the 2, and it's related to immune suppression. Again, here we're using MSIG-DB signatures that don't have overlapping genes with ours, and we see that this immune suppressed phenotype is related to immune suppression, in fact. That's how we unbox the machine learning, the neural network black box. So we can visualize the latent space. The x-axis is the output from the immune neuron, and the y-axis is the output from the angioneuron. The contours on this plot give us prediction probability that the sample belongs to one of the 4 phenotypes, as illustrated on this figure. When you observe or investigate data on this plot, right, you can make some treatment decisions. You would expect immune high patients to the right to be good fit for immune therapy. You would expect angio high patients to the top to be good fit for the anti-angiogenic agents, right? And the top right corner with high angio and high immune, that would be good fit for a combo therapy. In this case, we have gastric cancer patients that were selected for immune checkpoint inhibitor monotherapy. You can see most of the responders here to the right. The responders have green color. You can also see that response is the most enriched in the immune active phenotype to the bottom right. So there is where we have most of the responders, and if we were to choose this phenotype for patient selection, we would have a response rate of 58% compared to the standard of care that considers the PD-L1 biomarker and has a response rate of about 30%. When we look at the prediction probability of this biomarker, you can see it has bimodal distribution. It's a clear-cut separation between responders and non-responders. And when we compare that to traditional PD-L1 biomarker to the right, it just gives you better better choices for thresholding. We validated the panel under various conditions in various tissues for various therapies. In total, that's an old slide. It was validated on more than 10 cohorts and more than 5,000 patient samples under different disease modalities. We also looked at the prospective capability. We did most of our analysis in gastric cancer patients, but when we look at other tissue types, in this case melanoma and colorectal, we see that it fits our expected hypothesis that immune-active patients have the best prognosis in this case. Just to wrap up this use case, the XAIB biomarker now is in prospective clinical trial validation. What's also interesting to me is that it was developed originally on gastric cancer cohort, on microarray data, and now in clinical trials we have the same model but used in ovarian cancer and on RNA-seq panel. We'll just briefly look at the second case study for the sake of time, which is on KRAS-ID biomarker. We just launched this last month. The KRAS inhibitors burst into the scene last year. Treatment is assigned, as mentioned, based on the presence or absence of the mutation. The response rates are in the range of 30 to 40%. We can do much better when we look at the biology. Specifically, we looked at these 6 biology signatures listed on the slides, and those phenotypes were then included in the decision model. That's the overall schematic, just illustrating how the decision model for patient stratification is built on top of the foundation model, and it gives us response probability. When we take an inside look, right? This is how we could interpret individual predictions. On the slide, you see two patients in blue and in gold color. The patient in gold color would be predicted as responder just based on the biology, and the patient in blue, because of low activation of these biologies, would be directed towards alternative treatments. We did validation in preclinical data. Here I'm showing two of the axes, two of the biologies, because not all biologies are relevant in cell lines. They don't have the tumor microenvironment. You can see validation in 3 different drugs, 2 drug modalities, and when you look at the numbers, it gives you exceptional classification performance, AUC above 0.9 in all but one example here. We also partner with Tempus Therapeutics. We have real-world data of KRAS inhibitor treatment that was administered to G12C mutated samples. The black line shows selection based on the standard of care. We can do better, right? When we look at the complex biology, we can infer KRAS ID high subgroup and KRAS ID low subgroup. We can see that the KRAS ID high subgroup has twice or doubles survival time. Power calculations indicate that when using this biomarker, you need 4 times fewer patients in clinical trials for the same power. And since we launched this biomarker, we started collaborations with several big pharma and medium pharma companies, including Roche, Revolution Medicine. Just to conclude, the regulatory environment is ready to consider biomarkers based on RNA-seq and based on machine learning, but it's important that these biomarkers are explained in detail, interpreted, and validated on independent data sets. Thank you.
Okay, we have time for a few questions, and I've remembered the iPad. So there's some questions from people online. Are these neurons in the model feature slide from foundation models or a fine-tuned model?
So the neurons are features from the foundation model.
Okay.
Okay, but then on top of that, we have another model that's fine-tuned to patient outcomes.
Okay. Sorry, not my glasses. Sarah?
I was trying to get an idea as you were talking about this.
Sarah, could you mic just for people online?
Oh, sorry. Very interested in your biomarkers. You talked very little bit at the end there about the regulatory environment. And I was wondering, as we build these sorts of AI models for use in clinic, we obviously want to be able to update them. How is that going to work with the regulatory environment with such large data sets and constantly evolving ones?
That's a great question. The way it works today is when you update the model, you have to go through the whole process from start to finish. Through the Alliance for Artificial Intelligence in Healthcare, we work with government bodies on policies, right? So we are proposing a framework where you could propose a model retraining protocol that models could be retrained under certain conditions. And then also in advance, you would also propose validation procedure for the updated model so that you could get the updated model through the regulatory faster.
Dave?
Thank you. Could you tell us a little bit more about that UST? I think that model to the left of the final one you were talking about. Thanks.
So what we did was we aligned or we removed the batch effect with methods like combat. You probably know combat, right? But combat is so you have these population-based methods that can help you adjust the bias or variability between data sets. However, they are not really practical when you need to be able to map individual new samples into this universal space, especially when you don't know the original bias modality, right? When maybe you get a cohort with just a few samples and you can't use traditional population-based methods for batch effect removal. So what we did was we invested a lot of time into building this aligned space of all the available RNA-seq data that we had of hundreds of thousands of RNA-seq samples. And then we trained a neural network that maps raw gene expression into this aligned space, into the space where the variability is removed.
Unfortunately, we're out of time. There are another question, but if you've got them, please email me.
Yeah, absolutely. You have my email on slide. Thank you.
Okay, I'd like to thank the three speakers from this morning one more time. Please announce refreshments. There are refreshments. Please be in the next session for 11:15. Thank you very much.
and will be talking to us about enabling explainable and transparent AI in health.
Thank you. Thank you, Victoria. And thank you, Priyanka, and the organizers for the invitation. So yes, I am Francisco from Genomics England, and today I will give you a brief tour through some of my research in the area of explainable, trustworthy AI. I will start by introducing two key dimensions of trustworthy AI: explainability and transparency. Then I will illustrate this with two application cases, one of them using multiple types of biological data and network approaches to integrate them and make predictions of drug response. And the second application case will be the use of generative AI to support the curation of biological knowledge using large collections of scientific papers. And I will conclude with some takeaways and recommendations. So explainability and transparency are two complementary dimensions in enabling trustworthy AI. On the one hand, you have explainability that can be defined in different ways or approached in different ways. For example, you have model interpretability, which is about the understanding the decisions, the outputs of an AI system. We also have research in the estimation of feature importance that are relevant for predicting and validating key biological indicators that influence an AI prediction. You have other sub-criteria within this dimension. And then you have the dimension of transparency, which can be achieved through data transparency and quality. This means, in many ways, providing clear documentation of data sources, your data collection methods, your quality assessment techniques. You have also methodological openness that can be achieved by providing detailed descriptions of your models, including the model architecture, but also the development process and the types and characteristics of data used to train those models. You have other key aspects such as resource reproducibility, and of course, the ethical and stakeholder engagement that is required to increase or gain trust in these models. These two dimensions can operate independently, but at the same time, they complement each other because they can support, enable, or inform the other dimension. And these are the key topics of my presentation. I will next illustrate these two dimensions and how to achieve them using two examples. The first one is the application of network approaches for multi-omics data integration. In this example, you will see not only how different types of omics data can be integrated to make predictions with potential clinical relevance, but it addresses transparency by providing clear data integration methods and explainability by showing the complex biological interactions underlying those predictions. And the second use case involves the application of Gen AI to support biological evidence annotation that is important for supporting our updating, curation of our genome diagnostic panels at Genomics England. In this case, you will see that this approach enhances transparency by providing traceable sources of evidence and improve explainability by generating human-readable summaries of complex evidence that can be used by the human expert to make a decision on whether to include, to update the evidence connecting a particular gene variant with a particular medical condition. So the first example, multi-omics networks for differential drug response prediction. In this case, the goal is to integrate different types of omics data obtained from patients exhibiting different phenotypes. For example, types of disease, subtypes of disease, or different manifestations of a phenotype. So you may have a patient group A versus a patient group B that could involve different phenotypes, also different clinical responses or responses to treatment. And then the idea is to generate these different networks consisting of different types of data. So you have different layers of omic information for each patient group. And next, the goal is to integrate these these layers of omic information coming from the two or more patient subgroups into a integrated differential network. And then with this differential network, we can make predictions, differential predictions on this network. And this could be, for example, predicting the response to a particular drug. And then by doing this, we can use the network-based representations to explain how this prediction was made and the relevance of the data used for the prediction. And this is something that we develop and share within a single package, our package called Dr. Diamond. And this is a collaboration with colleagues in Germany and Singapore. So among the use cases that we investigated, we asked the question, can we use this differential network approach to estimate drug responses in cancer, in particular breast cancer, considering two subtypes of cancer that are clinically relevant, ER-negative patients and ER-positive patients. These are clinically relevant groups because they have different prognostic and drug response characteristics. So we use data coming from the TCGA, including the different transcriptomic, proteomic, and phosphoproteomic data layers. Also, we investigated the value of integrating metabolomics data. Then, in this case, for each cancer subtype, we built single-source omic networks that are here illustrated with different colors. Then, for each condition, we establish interlayer associations, that is, connections between the different omic data layers. Once this is done, we estimate a differential network, which is basically a reflection of the differences between the two patient groups. So once we have this, we can query the tool with a drug or a collection of compounds, and then considering the targets, the known targets for these drugs, we can make estimations of drug response, we can rank the drugs, and then we can investigate the underlying interactions for these predictions. So one key observation is that our predictions of drug response correlate with the known responses to these drugs using in vitro models. We use in vitro drug response data as a surrogate of clinical response, as an approximation, and we found that correlation. On the left side of this picture, you have just a plot summarizing the ground truth for our analysis coming for the Cancer Therapeutics Response Portal. So here, basically, you have a comparison of patient groups, a comparison of the cancer type groups, ER positive versus ER negative, observed in the cell line data. So as you can see, you have different levels of response defined or estimated using the full change in drug sensitivity. And then using this gold standard, we compare our predictions using the multiomics network approach. And apart from estimating the correlation between those predicted and observed responses, we can analyze the classification power using, for example, AUC ROC curves. And as you can see, there is we show the potential of this approach to make good prediction, especially when we focus on the top rank drugs for this data set. So apart from showing the potential prediction power of the method, we also illustrate how a network-based approach can be used to support the explainability of these predictions. So, for example, giving a drug like momelotinib, then we predict a drug response score, 0.63, which is relatively high in comparison to the other observed predictions. Then, after that, we trace the targets for this drug. In this case, you have JAK1 and JAK2. And then using this information, we can look at the differential ages for these targets in the differential network. So as you can see here on the left, for JAK2, there is no differential, a differential integrated interaction score, which is a reflection of how that drug is predicted to affect the reference patient group. But in the case of JAK1, you can see that for different interactions connected to the target, you have a stronger differential integrated interaction score. And that integrated interaction score is a reflection of the estimated strength of the interaction between the drug and these targets and these components in the reference network. So apart from that, we can continue going back into the network representation and say, okay, now we know that this drug is highly differentially, the network is highly differentially responsive to this drug. Can we go to particular patient groups and estimate the importance of the given targets for this prediction? And we can see here that in the case of JAK1, you have clearly stronger interactions in ER minus, negative, than in ER positive. So these plots can be used to interpret the predictions for a given drug, considering known targets. And this is consistent with evidence observed in the literature. And we have other application cases. And just the second part of my presentation is the use of generative AI for supporting diagnostic knowledge curation. The challenge here is to support our diagnostic pipelines to go from millions of variants observed in a patient's genome for a given condition, how we go from this amount of potential variants to fewer than 10 variants that can be used for supporting a diagnosis. So to support that, we have a knowledge base called PanelApp that maps gene to diseases using evidence coming from different resources, in particular the scientific literature. The challenge of updating these panels with the latest, most accurate evidence is high, is time consuming, is complex. So the challenge of, for example, for a given panel, for example, a panel describing associations between a disability, we have this evidence, supporting evidence criteria, green, amber, and red, with green genes representing those associations that are ready to be used in the clinic. So the challenge here is how we continue updating these panels using the scientific literature. So to support this task, we define a workflow that is powered by Gen AI, and the objective is to augment the curator's ability to extract, not only to identify associations, gene disease associations in the literature, but more importantly, to extract evidence supporting those associations so that the biocurator can update our panel app database. So here you have a description of the workflow that begins with searching for genes in the literature, then encoding those articles and sections of these articles in a format that is readable and useful for machine learning. Then next, we retrieve the articles that match a particular gene disease query. If the text is found in any of these articles, then we use that information as context to query a large language model to find associations and the supporting evidence. Then the outputs are provided in a user-friendly structure format to allow expert assessment and decision making. So I don't expect you to read this slide, but this is basically to highlight that the inputs, the query inputs given to the large language model includes a number of aspects and information that are relevant to obtain accurate and reliable associations and supporting evidence. We also ensure that these prompts induce the outputs that are also well structured and readable. And here's an example of the outputs where you have not only the prediction or the estimation of an association between a variant and a condition, in this case, intellectual disability, but also you have an enumeration of the evidence supporting that, and that evidence is structured into different levels of evidence. For example, evidence related to observations found in patients, observation related to different mechanisms, observations supported by functional studies in animals. And then, in the end, we provide a brief summary of the association and the supporting evidence. And this organization allows a fast and accurate assessment that is conducted by the biocurator. And here you have just a brief illustration of the kind of results that we have obtained so far, in which we have, in the case of the mental disability panel, we have been able to verify existing associations, but also upgrade or promote new associations that are suitable for clinical care. And beyond this pilot study that was done in cooperation with AWS, we are expanding not only the data scope of the system, but also we are implementing automated user-friendly pipelines to allow the users to have these outputs on a regular basis that can be assessed in an efficient way. So to conclude, my takeaways are that explainability and transparency are multifaceted and synergistic approaches. These two examples illustrate that interplay. For example, we can see that they mutually reinforce aspects in a given application that, in the end, enhance AI trustworthiness. This is observed also in the case of network approaches, where by revealing complex interactions, we boost not only explainability, but also transparency. In the case of the biocuration project, by doing this, not only we're accelerating the annotation process, but also improving the generation of human-readable summaries of evidence that are also useful for accelerating the biocuration process. And I thank my team at Genomics England and our partners at AWS, and also my collaborators in Berlin for this work. Thank you for listening.
Thank you very much. Do we have any questions in the room? So we have one here from John.
No methylation, but that's, the approach is, can be adapted to include other layers, definitely. But the methylation part, no, we haven't investigated that yet. In some cancer types, I would agree that this could actually enhance the prediction performance, in particular in cases where we know that methylation is a prognostic factor. But in the case of breast cancer, we didn't investigate that dimension.
Okay, I think we had a question in the row behind.
Hiya, thank you for your talk. I'm right here. Very quick and boring question. Which LLM embedding model did you use for the RAG pipeline, and did it affect your results very much? Because there are specific ones that are trained on or fine-tuned on PubMed, and there are more sort of normal ones. Is normal semantics enough to extract the summaries from each paper, or not? Thank you.
Yeah. So the embedding model that we're using, so we are using Claude model, first of all, within the Bedrock platform of AWS. So we are using the face model for indexing. And then we have, for the embedding, actually, we try different approaches. If I recall properly, the optimal approach is the one provided by AWS Titan, I believe it's called. So it's the AWS embedding model, and that's so far the optimal approach, and also the most efficient. Does it answer your question?
Yeah, thank you.
Okay, and we'll take one more question just over there from David.
Hi, thank you. And again, related to the RAG at the end, I was interested, you had Claude was returning a summary, and then, which I think you described it as like a chain of reasoning. Was that actually chain of reasoning? And how much do your curators actually, do they spot problems in that chain? I'm curious how often that happens, because you talked about trying to encouraging it to give something that was quite conservative. Also, on a separate angle, I'm going to squeeze in two, sorry, Victoria. How much do Tim Hubbard talked about the problem of reproducibility of research. Is there any chance that in using the LLM, you're amplifying existing fashion in science, and there's a bias there?
Yes, thank you. So the first question, the prompt that you saw there is just the output of a journey of prompt engineering, where the biocurators play a key role in providing feedback. So you mentioned a couple of points related to not only accuracy, but also the reliability of the outputs, including the consistency. So all these elements were used by the biocurator to help us fine-tune or engineer the prompts. So definitely that was part of an iterative process until we found a prompt that was not only structured enough to produce structured outputs, but also reliable enough and consistent enough. And the second question, I think that's a crucial question. In general, not only in the context of a literature review, I think it's one of the challenges for AI, how you reinforce existing modes of thinking and that rely on existing probably biased approaches to research. My hope is that by allowing the users to make the final decision and providing the best available evidence to support that decision, we can reduce some of those biases. Of course, that's not a guarantee, but I think it's better than just providing an output and saying, this is your prediction, make a decision, yes or no. So that's my current approach. I hope that's big. Okay, thank you.
Okay, please join me in thanking Francisco.
Thank you very much.
So just a reminder for those online, we're using Slido for Q&A, so please feel free to submit your questions online as well as the ones that we'll be taking in the room. So next up, we have Andre Altmann, who is an associate professor at the University College London, focusing on how molecular biology shapes brain function in healthy and diseased brains. And the talk today will be explainable models in dementia research.
Yeah, thanks a lot for the introduction, and thanks a lot for inviting me here to talk about my work. So I'm part of the UCL Hawkes Institute. If you have not heard of it, it's not a surprise because we had a soft launch this week. And we are a rebranding of two former institutes. One was the institute, the Center for Interventional and Surgical Sciences, so people doing integrated robotics when they operate on you, so a lot of robotics and AI. And where I'm part of is the Center for Medical Image Computing, what it says on the tin, everything that does imaging, we try to apply AI on it and try to improve things. So I'm working mainly on dementia, and as you can imagine, one key thing where we use imaging and AI is trying to do differential diagnosis. I'm saying differential because actually the task of deciding based on a scan if somebody's healthy or has dementia is not really a thing. It's a toy problem, but we're often more interested in figuring out what type of dementia you have and actually what's wrong in the brain. Of course, once you have a diagnosis, AI is involved in doing prognosis, integrating lots of different biomarkers, for instance, amyloid build-up using PET imaging, tau build-up using tau PET imaging. We try to do disease staging, where we integrate even more biomarkers based from blood, based from cerebrospinal fluid, cognitive biomarkers. And AI is involved in putting all of this together and then potentially help doing clinical trial enrichment, having more efficient numbers, doing risk stratification. And especially, I would say, we apply AI to try to improve the biomarkers that we have. So if you have imaging biomarkers, try to use AI methods to actually get the most out of those ones, reduce noise and transferability. So there's lots of options. So this is about explainable models. And just to motivate this, I brought a paper which is a slightly bit crazy. It is about using not so artificial neural networks to do medical imaging tasks, and it's deciding between breast cancer or not breast cancer in tissue samples. The setup is like this. I hope, yeah. So the authors essentially did this. They showed the pigeons those samples, and the pigeons had to decide left or right whether it's cancer or not. And obviously, if they were correct, they would get some food. So with some training, you could actually nicely see that over 2 weeks, they reach a reasonable performance of 90%, depending also on the magnification of the samples. That's pretty good. And they did not only memorize the thing, lo and behold, they actually did well on test data as well. So why are we not just replacing this with some MRI images? The NHS might be stretched, so it might be a good way to do that. So that's where we come to the key point of explainability. On one hand, we have an animal making decisions, which might be correct. On the other hand, we have a very highly trained expert who also has a trained neural network over a long course, but who actually is vocal and can actually explain the decisions and teach us on what is going on. So AI is somewhere in between those two things. And I think if you have a black box model, you're not much better than a pigeon, you're a bit maybe more reproducible. So colleagues of mine in the Institute actually devised a nice experiment, which I brought here, to open the black box. So this is work from Professor James Cole's group, the Manifold Lab, which is part of our institute. And in particular, it's Sophie Martin's work. She's a PhD student. And they designed a specific task to experiment with state-of-the-art explainability techniques. And what they used is, as a task, they had amyloid PET images, and they wanted simply to predict whether there's actually amyloid in there or not. Sounds a bit easier than it is. Then they had a data set which was collected around Europe with 180 samples, and all of those people were actually clinically non-demented, so they had cognitively normal. The labeling was provided through visual reads, which is pretty much a radiologist just looking at a scan and saying, yes, this is amyloid positive, or no, this isn't. And there were 34 positive scans and 166 negatives. And the way this looks, and the setup is pretty straightforward for an imaging setup. So on the left-hand side, you see the MRI PET image overlaid from a structural MRI, so that you can actually nicely see in which brain region it is. And so these PET traces have a lot of off-target binding, so you see a lot of things that are in the white matter that are accumulating, which is not amyloid, it just binds there. So it is not always that easy to decide whether it's an amyloid-positive scan or not. So Sophie fed this into a classic imaging setup, 3D, ResNet, and so on. And then she tried 3 different algorithms to open the black box. And the aim is, or the expectation was, that all those algorithms obviously should highlight the regions where there's actual buildup of amyloid that we did not expect. And so this is our gold standard here. The model did reasonably well, had a 90% accuracy. And you could say that, for instance, the method called guided backpropagation gives you a more or less fine resolution map showing highlights of where the method thinks on average there's amyloid buildup. That is another technique, occlusions. And you can kind of directly see that this is not very detailed, very patchy, not very useful for us. And there's guided GradCam. And you have now 3 options to do those things, and they actually look quite different already. And this is kind of the takeaway from this experiment. So there's especially guided GradCam and guided backpropagation here on the columns 2 and 3. They actually rely on biologically informative or relevant information, making the decisions, but they actually tell you totally 2 different reasonings. So there's not one ground truth that we can recover here. So it's not that easy. It's actually a difficult problem to move forward. But that was classic, I would say, trying to open the black box technique. So where the explainability is more of a side effect of what we try to achieve in order to build trust in the method. But there's actually other AI systems that also colleagues have been working on that are designed to look at the data that we have and explain the biology that's underlying. And one example of this, which I brought here, are disease progression models in Alzheimer's disease, for instance. So Alzheimer's disease, clinicians came up with this march of biomarkers. So we have a lot of different biomarkers in Alzheimer's disease, and just by observing patients, they came up with this theoretical model that, decades before you have cognitive problems, we have a buildup of amyloid, which saturates. Then afterwards, we have a buildup of tau. And then at the time when you become cognitively impaired, we have a shrinkage, measurable shrinkage in brain tissue. And then once you're demented, we have those characteristic memory problems and executive function problems. But that was a theoretical problem, so it wasn't recovered from data. And what colleagues of mine in the Institute, and specifically in the POND group led by Danny Alexander, were doing, they're specifically designing AI models to understand disease processes. And in this particular case, which is called the event-based model, they essentially were taking all those cross-sectional observations that we have typically in databases. So this process goes over decades. So often we only have cross-sectional data or maybe observations over 1 or 2 years where not much changes. And based on those, the aim was to recover the change of biomarkers. And you see along the diagonal which steps are following one after the other. A further step along the way is improving the model by assuming that there's not just one single characteristic transition of biomarkers. There's biological variation. So this model called SUSTAIN, subtype and stage inferences, combines this idea of estimating trajectories of the disease or of biomarkers, but also allowing for different subgroups of people, so biological subgroups of people being in the data set. So it's doing some sort of clustering and the stage estimation. And you can think of this if you're doing single-cell sequencing, where you have readouts, which are cross-sectional of single cells, and you try to reconstruct the developmental trajectory. So the nice thing is when you have these models, you can actually, when you've estimated, and you can link this back to genetics, and this is something my PhD student did a long while ago. She used one of those methods to create what in the bottom row is called disease progression score, and used it as an endpoint for a genome-wide association study. And we compared it to traditional biomarkers that we could infer from the images, like the size of the hippocampus or the amyloid, the amount of cortical amyloid. And this analysis showed that we could actually uncover a small peak in chromosome 4 around the L-CoR-L gene, which wasn't obvious for any one of the individual biomarkers. And the nice thing is that we could validate this in another longitudinal cohort, and showing that carriers of that variant actually have a slightly delayed transition to cognitive impairment. And by now, this variant actually is also known to be involved in Parkinson's. Another simple thing you could do, something that we actually did recently, is you have this very simplistic view of moving in the disease through stages. You have people who are with no biomarker positive, so there's a people with the 0 here, and then you move to amyloid positivity, and to tau positivity, and then to neurodegeneration. And this is a clinical tool that clinicians actually use to kind of have valid subgroups. And if you have longitudinal data, then you can actually link this back to genetics, and you can ask what happens actually, what are the drivers of moving people from no biomarker to the next step. And you can see that for people not having any abnormality and becoming amyloid positive, the key factor is actually the ApoE gene, ApoE4, as we've seen also in Tim's talk. It's a big risk variant. And a polygenic score for Alzheimer's disease, so all the other 70 variants, they're actually not contributing that much here. The picture changes quite a bit if you think of people who start amyloid positive, so when you capture them, and then you follow them until they're reaching the next stage or not. You can see that the impact of ApoE is actually much, much reduced, but suddenly all the other genetic variants play a key role in moving people who are amyloid positive to actually also become tau positive. So in the last bit of the talk, we'll focus on a model that is not explainable by accident, but explainable by design, and these are so-called normative models. And the idea of a normative model is that you try to model, for instance, the change of a biomarker with respect to a key variable like age. And many of you probably know normative models when you attend a GP appointment with your child that is newly born, where you actually have these growth charts where you have on the x-axis the age of the child, and on the y-axis body weight, head circumference, and so on. And GPs become, of course, a bit anxious and nervous if you're not on the curve or in the lowest percentiles, or if the child is not following the curve. So the same thing is something you can do now for brain imaging. And colleagues, or work initiated by colleagues here in Cambridge, have collected data on, I think, 100,000 brain scans, and it was published recently in Nature 2 years ago. And to build those brain charts across the whole lifespan, so from 0 to almost 90 years of age. And of course, for dementia, we are pretty much interested in the last bit of that curve. So what you can do now with these normative models in the clinic is something like this. So this is what colleagues at Quantitative Neuroradiology Initiative do. Since a few years now, they develop clinical reports that are centered around this idea that you could figure out which brain regions are unexpectedly small and use this for diagnostic purposes. So this is a dementia report where here in this traffic light system, you can see the measurements for the size of the hippocampus, and in this radio chart down there, it's actually different brain regions in the cortex and the subcortical areas that help clinicians making a quicker decision and interpreting this complex imaging data directly. These reports are now also available for epilepsy, also focusing on hippocampus here, but also for multiple sclerosis. These reports are great, but I think they ignore one key factor in variation, which is genetics. And this is work by my PhD student who recently defended 2 weeks ago. And he is integrating genetic information into those normative models. So he started by estimating a normative model on UK Biobank data, using Gaussian processes in order to get these nice probabilistic models. And then we looked at the GWAS that exists, for instance, for hippocampal volume. So, and the GWAS essentially studied lots of people who are healthy and identified, of course, a lot of genetic variants that increase or decrease the size of the hippocampus in healthy people. So now what you can do is you can generate a polygenic risk score for this, or polygenic score, and then split your group into people who have a genetically predicted larger hippocampus and a genetically predicted smaller hippocampus. And then you can build separate nomograms for those. And if you do that, you actually see, and then the black line is the ground truth, and the red line is for the high polygenic score group is actually shifted upwards, and for the low polygenic score group is shifted downwards. And it doesn't look like much, but actually for this age group, the shift between the high and the low group amounts to roughly 3 years of healthy aging. So we're going to add additional precision here to those estimates on whether you're abnormal or not. And Mohammed is now looking at how this affects estimates for cognition in people. So to this, he takes this model, which was trained on the UK Biobank, to a data set called ADNI, so it's an Alzheimer's neuroimaging data set. And we have lots of cognitive tests on those people. And essentially what he's doing here, he's asking a question for this table, so the significant value will relate to this idea, that we take a normal normative model, and then Mohammed asks the question, if I now do the genetic adjustment, are you shifted upwards, or are you shifted downwards? And we're essentially assessing the amount of the shift directly with how much it additionally explains your memory scores. And we can see that we get good performance for those memory scores at baseline, but most importantly, actually, we also get better performance for how the memory scores change over time. So we actually, by doing this additional adjustment, we actually gain additional precision. So Mohammed is doing this also now on another data set. It's called EPAD. It's a European data set which exclusively recruited people who are healthy but have a high risk of developing dementia. And at baseline, we see exactly the same pattern here. So the last 3 minutes, I'll walk you through an idea that we have, or something we're working on, where we not look at a single brain region, but actually at multiple brain regions, which we call multivariable normative models. And this is work by Anna, who also a few weeks ago or months ago graduated. And here the idea of a normative model is based on a deep learning architecture, so variational autoencoder. And again, the model, the autoencoder tries two networks. So you have one network taking your data, compressing it down to a latent space, and a second network taking that latent representation and unpacking it again to your target or original feature space. So if you train the model now exclusively on healthy data, for instance, in UK Biobank, you filter out everybody who's not, who has some disease, then this model essentially just understands on how healthy brains work. It's a bit like the Anna Katerina principle, where all healthy brains should look the same, whereas diseased brains should look different. So what you can do now is, if you take my brain, for instance, you can put it on the left-hand side in, you go through the whole pipeline, and then you compare the reconstruction to the original brain. And it shows you maybe that something is quite off and quite different, and you can quantify that. Or alternatively, because we have this variational aspect here, we have a probabilistic approach here in the middle, we can actually probabilistically assess on how far away you are you from the center of the data where you should actually be if you were healthy. Anna, since we worked on this, Anna actually improved the model to integrate multiple modalities. So with brain imaging, we actually have lots of different modalities. So this one is using structural MRI plus diffusion-weighted imaging, so it's a bit more fancy. And so Anna also took this model and trained it on UK Biobank, and then applied it to this EPAD cohort where there's only healthy people in there. And what we could see, or at least healthy at recruitment, and what we could see is that when we focus on this latent space distance measurement, we could actually very well tell whether people are still healthy, so with a CDR score of 0, or whether they have progressed to CDR score of 0.5. So the model is sensitive enough to pick up those subtle changes over time. The same is true for another cognitive measurement. Unfortunately, it's not really true for any genetic risk in this case. But the nice thing is, so where the explainability comes now, is that we can go back to this input-output question and ask specifically, are people showing massive differences in specific brain regions? And when we do this for the people that show clinical dementia score rating of 0.5, we see that there's shrinkage in the hippocampus, these slight blue regions. And the same is true if you do this for this group that has slightly decreased memory scores. Again, it explains us really where the damage has happened to the brain without trying to go through the backpropagation process. And we also see some differences for homozygous ApoE4 carriers. So to conclude, explainability, I think, in imaging science is very important to verify the biological basis of the decisions. And the other thing, the strong suit, is that we directly build AI models that learn and explain our biology. And by doing this, we actually have a nice way in creating new biomarkers that we can study with genetics and understand even more of the disease. But last but not least, I would try to emphasize that normative models are a way of directly building explainable models and not just having them trying to reconstruct where the decision came from. For this, I'd thank my team, so lots of alumni here, collaborators. Questions? Thank you.
Thank you very much. Do we have any questions in the room? Got one just down here. Just wait for the microphone to come down.
Thanks very much. Hi, thank you very much for the talk. It was fascinating. I was wondering, with your VA model, what were the covariates? What was the nature of the covariates you were using through the model to provide additional information?
So in that case, things like intracranial volume, gender, age, these kind of stuff that we're trying to, essentially we try to make it independent of the latent representation, independent of those covariates.
And did you see that those, when you ran the interpretability methods back through to look at different brain areas, did you see any strong interpretability scores for those covariates through the middle as well? Were they providing strong influence on the predictions?
Pardon?
When you ran the interpretability methods back through the VAE for visualization, did you get any strong interpretability or attribution scores for the covariates as well?
Yeah, we didn't run the classic guided backpropagation back through the normative models. So that's really just the difference between input and output that we try to highlight and measure the amount of how much different it actually is. Yeah.
Thank you very much.
Okay, we have time for one more quick question.
Hi, thank you for the talk. I was wondering, so if we're comparing sort of the normative models against the taking a black box and then trying to reconstruct a decision post hoc, do you see that, do they answer different questions, or is one preferable to the other in most cases?
Yeah, that's a good question. So, I mean, I glossed over this, but of course the normative model does not give you, so I mean, we have two ways of doing this, but if you just grade specific brain regions, whether they're abnormal or not, you leave the decision essentially with the person looking at the report. You could, of course, train another model on top of this, where it takes this fancy Z-scores, you could take, fancy Z-scores, and makes a final decision, and then you could look at the weights. But yeah, they're serving a slightly different purpose, whereas I think the normative models are more acceptable in the clinical settings, because it's something, you know, we have normative values for blood pressure, we have normative values for everything. So it's an easier transition in, I think.
Thank you.
Okay, please join me in thanking Dr. Altmann again. So moving on from the disturbing proposition that pigeons could replace us all, we're going to move on to Zoe Kourtzi, who is Professor of Experimental Psychology and Computational Cognitive Neuroscience at the University of Cambridge, to talk to us about AI for better brain and mental health, from cloud to clinic.
Thank you. Thank you very much for the invitation. It's really wonderful to be here. And thank you, Andre, for a great introduction. No pigeons, sorry. Just really to state the problem, as you've heard in previous talks, looking into brain diseases, it's really critical in terms of societal benefits. We know that 1/3 of the global disease burden comes from neurological and psychiatric disorders. We have experiencing a serious mental health crisis, especially in young people, and dementia is hugely costly, huge societal burden, and the primary cause of death in the UK. AI and machine learning provides us really with tools to be able to make interpretable predictions for brain diseases. And I wanted to tell you a little bit about this kind of success story in the space of dementia, linking also to Andre's talk. So what are the challenges we have? First of all, one key challenge with dementia, except of course of the very high costs and the fact that currently 55 million people worldwide live with dementia, is the fact that dementia is actually very heterogeneous. So we don't have one type of dementia, we have many different types of dementia, and we also have people that present with comorbidities that show very similar symptoms to dementia but don't have dementia pathology. So when we try to diagnose dementia from the first symptoms, we actually know that we have up to 30% misdiagnosis. Now this is really huge for healthcare systems, it's huge for patients. Patients typically at the initial stage, when they experience symptoms and they'll go to their GP, they will have some basic kind of cognitive test and maybe an MRI. And if there are very early stages, most of the times the diagnosis is not conclusive. And then they will be sent forward for more invasive tests, for example, a lumbar puncture or a PET scan, which are highly costly, highly actually invasive and burdensome for the patients. And the patients effectively go on a trajectory which is quite anxious. Now, if this patient does not have dementia pathology, but they have, for example, cardiovascular disease, diabetes, or they have depression and anxiety, they might be experiencing similar cognitive symptoms like memory loss or confusion, but they don't have dementia. Now, their symptoms are only going to get worse if they are going down a very lengthy, burdensome, anxious pathway for them. And of course, the NHS is investing a lot of resources to people that might not actually have the disease. Now, to make this even a harder challenge, so far, for about 30 years, clinical trials targeting dementia pathology had been unsuccessful, one after the other. Only the last 18 months, actually, we have seen 2 monoclonal antibody drugs that have been successful, and they show changes in cognitive symptoms, up to about 27%. Now, these drugs, one of them has actually just been approved by MHRA, not nice yet. They're hugely expensive. In the US, they cost $8,000 a month, and they have severe side effects. They can cause death due to severe neuroinflammation. So who do we give these drugs? When do we give these drugs? There are 127 targets currently ready to be tested. How can we make our trials more efficient, cheaper, faster, and with better outcomes? So this is really the space where we strongly believe that machine learning can come in and help us to address all of these challenges. And today, that's what I want to show you, how we can build models that they are transparent, they are interpretable, they are robust, and they can be translated to clinical practice. So you've seen this picture, Andre had it, is kind of like the holy grail of dementia research. What I want to emphasize here is that the earlier we are trying to predict dementia pathology, the least predictive ability we have, because the tools we have are not sensitive. If we want to make a difference in patient well-being, in drug development, we need to predict early. If we predict late, we have huge neurodegeneration in the brain, and therefore we have in our hands a very different problem. We have a brain repair problem, which is a very different ballgame. If we want to make an intervention that is successful, we need to be early. We also know that changes in the brain that might cause dementia start about 10 to 15 years before we have symptoms. Now, this is a golden window of opportunity to intervene. If we intervene early, we can change 40% of dementias just simply by lifestyle interventions. We can change the pathway of the disease. We can have better treatments for patients. So it's really important to intervene early. But the problem we have, early we don't have good markers. PET scans are unlikely to show anything. MRI scans with a naked eye again, unlikely to see anything. Blood markers that are now coming into play potentially can help us here. And here's exactly the problem. So as I said, currently an individual that experiences symptoms will go to their GP, they'll have a basic kind of cognitive scale run into them. If the scores are looking low and there is reason for concern, they get referred to a memory clinic and they will have an MRI scan. Now, if looking at this MRI scan, you can see that there is atrophy in the brain, there is lots of CSF, large ventricles, therefore there is reason for concern. This actually is a brain of an Alzheimer's patient. But if I show you these two brain scans, it's really difficult to tell which scan is from a healthy brain versus a brain at early stage of disease, what we call mild cognitive impairment, or MCI. And actually, if I didn't have my notes, I wouldn't know. Either the one on the right is the patient that is at early stages of dementia. So this is a very difficult problem for clinicians, and that's why we bring in machine learning models that we built to be multimodal. At that very early stage, no marking can give us a reliable, robust, conclusive prediction. Therefore, we train the models with thousands of data from research cohorts, including many different modalities. So we include genetics, we include MRI, we include biomarkers from PET scans, amyloid and tau, accumulation, we include cognition, and now we've got blood markers that we can also include in the models. And we built architectures that ensure they are transparent and clinically relevant. So the very first step of the architecture we make is we test the markers that we put into the model to make sure that they are clinically relevant. I'll show you how we do that. Then we go through a basic classification problem that is actually quite subtle and quite precise. We ask the question not whether the models can separate healthy from dementia, but actually whether the models can separate individuals that will remain stable in their brain health trajectory or they will progress. They will move away from healthy aging. And finally, we turn to these models into trajectory predictions. We are interested about stratifying the patients based on their prognosis rather than based on what happens now. And the reason for that is because we are dealing with neurodegenerative disorders, which means we are interested to understand how people progress, how quickly they will progress, or maybe whether they will remain stable. So this is the type of architectures we built, and I'll show you a little bit about the model. So starting with predictors, as I said, we make sure that they are clinically relevant. So for example, as I said, we give the models many different data. An example is MRI scans. The first thing that we do is we look what features actually we can extract from the models. Here is a simple example where we start with structural MRI. We do a PLS regression on the MRI images against changes in cognition. Effectively, we ask the question of what are the features in the brain that predict cognitive decline. And it's no surprise, actually, that the features we find are around the entorhinal cortex. Now, when I showed this to clinicians, they kind of went, well, yeah, what did you expect? But for us, this is the first breakthrough, because this means that our models then can rely on clinically relevant features. And it's interesting that the features we find are not in hippocampus. They are in entorhinal cortex. We know from research that in entorhinal cortex, we see the very first changes because of dementia pathology. So when we look at that very early stage of we try to predict dementia pathology at early stages of disease, these features are highly clinically relevant. Then we move into looking at this kind of hidden space of the models, and we ask the models to separate the patients that they will remain stable in their trajectory compared to patients that they will progress. And this is a very fine kind of discrimination. Depending on the data that we will put in there, our models can get up to 92% with sensitivity and specificity. And effectively, what we build is through metric learning, we work in a space where we look at prototypes. We have two classes to start with, stable versus progressive, and we look at these two prototypes, and we calculate then the distance between these prototypes based on a scalar projection method. So what we ask is effectively, if one prototype is the stable prototype, any new patient that comes in, it gets compared to that prototype, and we ask, will they remain stable, or will they start moving away from that stable prototype towards disease? And effectively, what we can extract through this is a score from this modeling architecture that tells us where a new patient stands compared to healthy, stable cognition, and how fast they move away towards disease. So the higher the score, the further, faster the patient is moving towards progression. Now, how do we validate these models? We have individual scores per participant. We cross-validate this within cohorts, research cohort data. Andrew mentioned it. But we also looked here at many different data sets. Dementia is a global problem. We cannot focus just on data from one region. We need to be able to ensure that we validate this across many different populations. We're still working in this space, but so far what you see here is data from many different cohorts that we have looked at. And we pull the clinical labels that we haven't given these labels to the model, but we look at the scores that the model has given us and how they relate to clinical diagnosis. And what you see here effectively is patients that already have a diagnosis of Alzheimer's disease, the model primarily moves into the rapid progressive space. This is all done based on normative data in terms of we can now split, sort of bin these scores into stable versus progressive, slow versus rapid progressive. Patients that they have no dementia, the model primarily puts into the bin of stable. So this is kind of a first validation based on clinical labels, which the model has not seen in the training phase. Now, the most interesting thing for us is actually the bottom kind of part of this graph where it says pre-symptomatic. These are individuals that have volunteered for research but have no symptoms. They've undergone through the same tests that the patients have undergone. And what you see here is some of these patients, or most of them, fall under the stable space. Their scores are low. Potentially, they will remain into having healthy progression, no decline in terms of pathology. But some of them fall into the slowly progressive. Now, are these the people that we want to be following early through brain health checks? Are these the people that we want to be putting in clinical trials for developing targets that allow us to intervene early? And these are really hot kind of an important translational questions that we are hoping to ask. But of course, to be able to move these models to the clinic, they need to be trusted. And we keep working on building these models to be interpretable. As I mentioned, the first thing that we do is we ensure that all the markers, all the features, the diet of these models is clinically relevant. And we believe that this is actually really important for these models to then generalize to clinical data, and I'll show you a little bit of that. Then what we do is we open up the metric space, and we look at the weights. We look into how much each one of the features that we gave the model contributes to the model decision. And what you see here is the kind of hottest the color, the highest the contribution of each one of the features to the model prediction. Not surprisingly, amyloid is a quite good predictor. We know that it's a risk predictor rather than a, if you want, diagnostic measure. But you can see also that gray matter and APOE4 contribute. And the same thing, these are cognitive scores that contribute to the model. So that's one way to understand the interpretability. The other way is when we have the scores of the model, we ask how much they relate actually to clinical markers. So what you see here is the higher the score, the higher the decline in terms of cognitive decline. The higher the score, the higher the amyloid accumulation in the brain. The higher the score, the higher the atrophy in the brain in terms of reduced gray matter in the brain. And also the scores relate to APOE4 positivity. So these are clinical markers that we can validate the scores of the models against. And again, that increases the interpretability and the clinical utility. So the final thing that I want to show you is how we can now take these models and translate them to the clinic. And we can ask really questions of how we can change the clinical pathway. So if clinicians have the scores of these models for every new patient that comes to their clinic, and they have normative data, they can bin the patients, they can change the diagnostic pathway by looking at who is going to remain stable and who is going to progress. And this can be really a transdiagnostic approach. This is an approach that is looking on how we treat symptoms. So if somebody is stable, they can go home. If they are slowly progressive, potentially they need follow-ups. If they are rapidly progressive, they need immediate care to avoid, for example, other conditions that might accelerate hospitalization. Now, how do we know that this will work in the clinic? We took this model already trained as a trained engine, and we tested it against clinical data. We built it on research data, and now we validate it in real data from real patients that come from memory clinics. Now, this data they're much more broader in their demographic. They are much more missing and messy because it's everybody who went to their GP and got referred to a memory clinic. This is data from the Addenbrook's memory clinic. You see these are the scores of the patients, these are the clinical diagnoses that the patients were given, and you can see that when we apply this model to patients in the clinic, patients that already had a diagnosis of AD, the model says that they are already progressive and fast progressive. Patients that they have other cognitive disorders, functional attentional disorders, fall in the stable bin, which means they don't have dementia pathology. We did the same with data from Singapore. Singapore has a great digital database because they've been developing this for a long time. And you see again here very similar results. Our models are trained with US data. They predict about patients in Singapore and patients that already have the AD diagnosis. They are already in the rapidly progressive space. Patients with mild cognitive impairment are more kind of in the slow progressive, cognitive normal in the stable space. Now, the question that I'm always asked when I present this data is, okay, if you move this into clinical practice and a clinician has access to this tool and they're looking at these scores, how do they know that actually these are validated longitudinally? How do they know that the model makes a prediction today from that very first visit and assessment that the patient has about what will happen to them in the future, and that is valid? Is it ethically valid to give this, to use this for diagnosis in the clinic? So we looked at this data from Singapore. They had data for 10 years. What you see here is about 6 years in terms of that's where we have most, the highest bulk of the data. And what you see here is the model predicts at time 0. This is the very first assessment. And in fact, the model can predict only with clinical data. This is cognitive scales and an MRI scan. And if you take the prediction here of a clinician at the same time and you project what happens to these patients in the future, this will be what the standard of care is. Effectively, this is what we do now. A clinician makes the diagnosis at the first assessment of the patient, and then we can see what happens to them in the future. Some of them decline. Now, what we found when we applied the model is that actually 50% of the patients in this sample were stable. They remained stable. They didn't show any changes, any decline. This is 50% of people that, if we applied the standard of care in terms of the current diagnosis based on a clinician's diagnosis, they would have gone through for PET scans and lumbar punctures because they were diagnosed as having mild cognitive impairment on the trajectory for dementia. Now, 15% of them have a massively, drastically different trajectory. 15% of them are rapidly declining based on the model prediction, and 35% of them are slowly declining based on the model prediction. This is the longitudinal validation. We are very excited about this because you can see at 18 months' time already, these curves separate, which means 18 months' time, you will know whether you made the right predictions. 18 months' time, a clinical trial should actually show changes if the right people go in. And that's the final thing that I want to show you. We have trials ongoing, trials that have failed. We need to do something about the efficiency and the efficacy of these trials. And we can do this by changing the way we stratify patients. Currently, trials will recruit from this MCI space, the mild cognitive impairment space. But if we have more precise stratification, for example, with these models, people who are stable should not go into the trials. If they go into the trials and they get randomly kind of put into the drug group and the placebo group, simple statistics, this is not going to show any difference. Stable people could be screened out from the trials. And then who are the people that we put in, the slowly progressive or the rapidly progressive? It might depend on the target that we have in our hands. And just to show you quickly, we worked with a pharmaceutical partner. We looked at a trial that failed. It was a trial that targeted clearance of amyloid. When we tried to stratify the sample that they had based on APOE4, you see that it's a cloud. We cannot separate it. When we ask the model to stratify the sample, you see that it can separate nicely, slowly progressive versus rapidly progressive. Now, I can't show you the data, although I have now approval to present it in 2 weeks' time at the Clinical Trials AD conference in Madrid. But what I can show you is progressions from power calculations. If you put in only the slowly progressive to a trial that looks at reducing amyloid, you see that you need about 200 people in your placebo and 200 people in your target group to actually see a drug effect, while if you're putting in rapidly progressive, that will completely ruin your trial. So to finalize, I hope that I've given you some evidence about the potential really that we have with machine learning models when we make them robust and interpretable for these models to generalize into clinic and into clinical trials. I'd like to close by thanking a fantastic cross-disciplinary team of engineers, clinicians, computer scientists, mathematicians, neuroscientists, and everybody who gives us the funding to make progress in this work. Thank you.
Have we got any questions in the room? I had a quick question about data sets.
Yeah.
Are there any limitations to the data sets that you can use, and are there enough data sets out there to expand this?
Yeah, so when we started this work, it's about 10 to 12 years, actually the data sets were very, very limited. You might have noticed that I talked broadly about dementia, but actually all the slides talk about Alzheimer's disease, and that's because Alzheimer's disease is the most prevalent type of dementia, and that's where we now have more data. There's been a big push for people to open data. It's a huge plea to everybody, clinicians, pharmaceutical industry, to open data. We have secure data environments now that we can afford actually to safely work on data, and that can make a huge difference. So access to data can be a huge limit in terms of how robust and the validation of our models can be, but we also know that there are huge differences across populations. So data that we have worked with come from the US, come from the UK, come from Europe, come from Singapore. We now know that populations in Africa could have very different genetics, potentially different brain features. We need to be able to understand all of this, and we need to ensure that then the models we build take into account this variation, of course, in demographics, socioeconomic status. All of these make a huge difference. So it's a very important factor for building models that are validated and generalized, that then can make a difference in real life.
Thank you. Any questions in the room? Yeah. Aha. Perfect.
So you mentioned that you use clinical features to train your ML model. So as you're going in progression, as a patient is going in progression, how do you make sure the ML model is being trained and learning through the other features, maybe due to environmental factors or anything, or age reflection, because you're also studying neurodegenerative diseases? How do you make sure?
Yeah, that's a good point. So there are some features that effectively we put into the models as regressors. So we want to make sure that differences in age, in education, socioeconomic status, sex, that's not what the models learn. And effectively, we will regress them out from the features that we put into the models. Now, we train these models with data at time zero in terms of the very first assessment that patients have, or the very first kind of baseline assessment that we have in a research cohort or in a clinical trial. And then we make predictions about the future. And when we make those predictions about the future, we look into trajectories. And therefore, we validate this prediction at time zero, at the very first assessment, against longitudinal data. We are working now also with models that actually what they model is the exact trajectory, and Andrea showed an example of these event-based models. We've built these mixture state models that they take the whole trajectory of patients. These are actually quite powerful models because they contain more information about the progression, and we can turn them into being unsupervised. So this means they don't need any clinical diagnosis. We can apply them very early to data before people have any symptoms or any clinical diagnosis and make predictions about their health trajectory in the future. So in these models then we take into account actually the longitudinal data. The models learn from dependencies in time.
Thank you.
Yeah.
Okay, join me again. I'm waiting to get the look on time. So if we can hold questions till over lunch, can we thank Zoe again for a brilliant talk? Okay, so our final speaker of the session is Deogratias Mzurikwao from MUHAS Tanzania, joining us virtually to talk about the role of explainable AI in biology.
All right, thank you very much, and thanks for having me. Apologies, I couldn't make it, although I wished to attend this conference physically. So I'll just talk a bit. It was a pleasure being in the UK for the past 4 years, because that's where I did my PhD in the applications of AI in healthcare. And I'll speak a bit on the explainable AI, its role, especially the challenges that we are having, especially here in Africa. Okay. All right, so since I came back home 3-4 years ago, I've been trying to pioneer the importance of responsible AI, because we have very few universities that they are offering degrees in artificial intelligence. So most of the practitioners here actually learn about AI from online resources, where there is very little information on ethics, where AI can go wrong, because every time when you go online, you see how amazing, how great AI is. And us as researchers, most of the time, we don't publish papers when AI fails. So most of the kids here who are teaching themselves on AI, they don't look much on the side of where AI can go wrong. So basically, the responsible AI comprises of 4 parts. To underscore fairness, I think for quite some time we have been hearing some algorithm being biased. Also, transparency and explainability, especially in healthcare. When you go to see a doctor and you are diagnosed with a particular disease, I think you have the rights to know why a doctor reached to that conclusion. But for quite some time, people have been calling AI black boxes, especially the deep learning algorithms. Yeah, and also privacy and security. How did you access the data? Just a very basic thing. 3 years ago, I received the data from a colleague who forgot to remove the patient's name and their social security number. She didn't know by that time on how much you can play with that data. So it's something which we really need to look into, especially when we want to create algorithms which are human-centered. So why do we need explainable AI? For a diagnosis, when something goes wrong, at least you can take the hospital or a doctor to the court. But what will you do if an AI algorithm goes wrong? Is it the developer of the algorithm, the person who collected data, or when a machine, maybe there's an accident at a factory, we actually know it's the fault of the machine. But when we come to AI, it's a bit challenging. So we have to make sure we develop this explainable AI. So this is an example of an algorithm which was trained to identify a train. But and it was a neural network. But when later on, we look back into the train model, the model was actually not looking at the train, but it was focusing on the railway tracks. So try to imagine you put this kind of model into a self-driving car, the car will stop forever waiting for the tracks to move, thinking it is a train. So this is just a basic example on how explainable AI can really have a change. And also there are some failed AI projects. So I tried to collect some of the AI solutions that have been created by big tech companies. Luckily, due to the regulations in their respective countries, at least these algorithms were removed. This is Microsoft Tay. It was a chatbot created by Microsoft some years ago. Later on, they came to learn that the algorithm was biased towards men and rejecting, it was using abusive language against women. And this one is an Amazon recruiting tool. As we all know, the size of the Amazon company, to review all the applicants was a bit challenging, so they created an algorithm which was able to review these applications. But later on, they came to learn that the algorithm was giving favor to male applicants more than female. So at least it was removed and they stopped using it. And this happened in the UK. This is a football club in Scotland, and I was just checking their performance. I think they're in third tier, where they created the algorithm to track the football. But later on, they came to learn that the algorithm was focusing on the bald head of the referee, not on the football. So these are just some of the examples, Uber self-driving cars and this kind of thing. So this happened to the global north, where regulations are there. What will happen to developing countries like Africa? It will be a disaster. And in healthcare, that's someone's life. And the list goes up and up. So most of these mistakes can happen during learning of the model when we are not using the right performance measures of the algorithms. This can also happen when we are collecting data. Yeah, and my personal experience on explainable AI, I will explain a bit on this one. I was working on a brain-to-computer interface application, and by using basically, this data was collected in China. I did my master's in China. So where amputees, where 4 amputees who have lost their limbs, their hands. So they are being asked to imagine with motor imaginary activity, to imagine that they're performing a particular activity. And this, it was a 64 channels EEG device. We are not expecting to see someone wearing this kind of device when they want to control their prosthetic arm. So using explainable AI, we managed to understand exactly the activities were happening on the brain. That means we can use small sensors, and this sensor can be placed right on the area of that individual instead of wearing these huge sensors. And this one was published in Nature using visual evocative potential, in which we came to learn that most of the activities were happening at the back of the head, and we managed to create a skin-like sensor, and these individuals were able to use the sensor for 2 weeks, and were able to actually take a shower, and it was easy to place the sensor at the back of the head. This paper was published in Nature a few years ago, and the sensor was patented. Back home, 2 years ago, we were approached by UNICEF, we were approached by UNICEF, in which they came to learn that children under the 5 years of age, most of them were not completing vaccination. And nobody understood what was the reason, but one of the basic concepts was probably due to socioeconomic status of the individuals. That's why they never get back to the hospital. Now that challenge comes. In order to know someone's socioeconomic activity, there are some BHS questions, which I think there are 49. But it is impossible to ask a mother with a baby at the hospital 49 questions while the baby is crying after injection. So the UNICEF approached us to see if we can create an algorithm that can lower the number of questions, but still be able to get the socioeconomic status of an individual. And we did that project, and it actually worked real well with a specificity of 94.2, and that algorithm has been deployed at the health clinic here in Tanzania. We're also supporting the Tanzania Ministry of Health, which they are using AI for screening of tuberculosis with explainability. And this is the only AI algorithm registered in Tanzania. And that's one of the challenges there, that when we were doing registration of the algorithm at the TMDA is Tanzania Medical and Drug Authority, their facilities was not ready to register AI algorithm. I remember one of the questions which we were asked is, software is being treated as a medical device. And one of the questions was, we are supposed to provide dimensions of the room in which this algorithm was created. But we all know that AI algorithms are being created in the computer. So you can actually see even the regulators themselves, they don't understand, they have very limited knowledge regarding AI. So it's still a challenge. And another challenge is on the data. So allow me to, in order to address the challenges that I mentioned, allow me to introduce you to ETH Lab, the lab which we managed to establish 2 years ago. We are calling it ETH, Emerging Technology for Health Research and Development Laboratory, where our main aim is to research and develop healthcare solutions. And this laboratory is under the university, and we are lucky that within the laboratory, we have some GPUs to train these AI algorithms. And one of the challenges that I mentioned earlier regarding data, now we are working closely with the hospitals to make sure that we collect good quality data from the hospitals. Because one of the things I came also to learn, it was just last year, I wrote a proposal to develop an AI algorithm for early detection of a condition called DCM, dilated cardiomyopathy. By the time when I was writing the proposal, the hospital told me they have data from more than 10,000 patients. When I secured the funding, going back to the hospital, there was only data from 1,000 patients. They deleted the rest of the data because they don't have server. So most of the time in our hospital, they delete that data. So this lab is trying to address that challenge. And you can see one of the GPU that we are lucky enough to have at the hospital. Sorry, at our lab. And that's how our lab look like. Looking for collaborators that we can work together, data sharing, building this algorithm and testing them. And these are team members from our lab. And we have another number of ongoing projects that we are working on for early detection of breast cancer funded by this one was funded by Google. We're working on algorithm for early detection of rabies epidemic funded by Lacuna. And also we are working on another algorithm for early detection to predict the outcome of sickle cell patients who are using hydroxyurea drugs. This is funded by Novartis. We're also working on algorithm for early detection of cervical cancer, and all of these data are local data sets, so we are trying to address these challenges, as even the previous speaker mentioned about the challenges of having data from low and middle income countries is very challenging, and most of the time, even when you have access to data, the quality of that data is not ready for AI machine learning, and this lab is trying to address these challenges. And here are my contacts. Thank you very much. Welcome for questions.
Thank you very much. Do we have any questions in the room? John? No? Oh, microphone on its way down. We've got one question in the room just coming.
Hi. When you talked about responsible AI, obviously getting all the criteria you want in the convoluted deep learning model just doesn't seem possible. Are you saying for that level we need to do things like build explainability from the beginning, like from the bottom up in our networks, or is there a way of using deep learning architectures to get to our AI?
I can say as long as we manage to make these algorithms explain themselves, like give the actual context, I think both ways. Like recently, I've been seeing people using large language models to even explain the result from the algorithm. So for me, I support it anyway, whether from the bottom or using heat maps or using natural language processing. So I support both.
Okay, any other questions in the room? In which case, join me again to thank Deogratias. So I now have the pleasure of announcing lunch. I'm currently standing between you and it. Just before we go, we'd like you on your phones to complete a 70-question survey. I'm kidding. You're all free to go to lunch. And I think, is it 1:20? 1:30 return.
Okay, I hope you can hear me, and thanks very much. I wish I could be there. I used to work across the street from the building you're currently in at the EBI as a group leader in research, and also a special shout out to Tim Hubbard, whom I met at the time, and others whom I know in the audience. And I want to tell you about some recent work that we've done that relates to the risk of cancer. And this is an effort that tries to catch cancer early. And the way we do that by looking at clinical records from patients to then predict the risk of getting cancer before they get cancer. And with this, of course, you have to train. If you're successful, you can actually design surveillance programs where people are looked at in great detail, and then early intervention to actually do something about the cancer before it gets too bad or even before it arises. This was very much a collaboration, coming together different institutions, both from Copenhagen and Denmark. That was very important. And then the Cancer Research Institute, Dana-Farber, Veterans Administration, MIT, Harvard, and funded by philanthropy, and also another contribution from Denmark, from the Novo Nordisk Foundation. The challenge is that pancreatic cancer, such as well as ovarian cancer and others, are very aggressive. They're detected, typically 20% are detected in early stages. You see that on the left. And those actually have a much better survival, 5-year survival. But most of them actually have a survival that actually is abysmal. And therefore, the point is, if one can actually detect the risk of cancer early and do surveillance programs with new technologies to detect the cancer early, like from blood samples, like some of the recent work from the UK Biobank that you may know about. And then, of course, one can actually resect the cancer and move more patients into an area where they have better survival. So catching cancer early is the goal, and that very much has to have clinical implications. To actually successfully reach the clinic, you have to not only have risk prediction, which is what we worked on here in the first step, but then you have sensitive detection methods, for example, from bodily fluid, which would be ideal, or some imaging methods, which can be expensive. And then you have to intervene by either doing early therapy, could be surgery, other kind of therapy, or even better yet, prevention of cancer for those people who are at high risk. That's the clinical application. We tested that by accessing very large electronic health records, as is essential to be trained, partly to cut through the noise in clinical records. In Denmark, 6 million US Veterans Administration, which is one of the best organized in the US, which otherwise is very fragmented, 10 million patients. So we train on a lot of patients, a small subset of which have the particular cancer, pancreatic cancer, and then you have to test this on withheld data. The databases are essential, and it's very interesting and complex and challenging that the distribution of in terms of trajectory length and also time of availability of clinical records actually is different in different healthcare systems, so that becomes an issue. So the data quality matters a lot, and you have to have an access to a confidential behind firewalls, a GPU cluster, and that's a challenge that has to be solved. There's one aspect of getting to clinical applications. The machine learning you're all familiar with, let's show briefly. The innovation that we used, other than beyond well-known AI methods, is to use time trajectories of patients' disease, one of those 6 or 10 million, the actual history going back some years, not just whether or not they had a certain kind of disease. And then when you make the prediction at a certain time point before they have cancer, to actually predict whether or not they're going to get cancer within 6 months, 12 months, or 3 years. And so there are 2 time aspects to that, and that affects, of course, how you do the machine learning. The analogy is natural language, where you have a system of words, and then you try to see what comes next. So similarly, disease events, this is a time axis, the black bars are disease events, the question what comes next. And we formulate that because of the time element that we want to capture as a probability curve over up to 5 years or 3 years, what's the likelihood to actually get cancer? No cancer, 0, 0. And then in the third year, somebody got cancer. So we train on data that is binarized in this fashion, but over a time sequence. And that is quite beneficial in terms of designing an intervention program. It takes into account whether or not somebody is at risk of getting cancer very quickly or over a longer time plan. The training is done on these large databases, and as you're familiar with, you have to have a training set, maybe a development set to develop optimized hyperparameters, and a test set that's withheld, you never look at least in theory, you never look at it. It's all withheld, and you do tests on that. Retrospectively, you test on that withheld data set. Ideally, you would test those methods prospectively, but this is the best way, a withheld data set, to test whether or not something actually is working on the data system in which you've trained it. And so what we're using is clinical histories. These green circles are disease events, cancer occurrence or not. And so these time elements are taken into account. You put that through a transformer, and then you optimize a loss function, which here is the difference between the observation, which is binary, and the prediction, which is a slowly rising probability curve, so that this gray area is minimized. So that's how you train. It's supervised training. And the key element after transforming things from a discrete state into real number vectors is this encoder. And even though cancer diagnosis is a binary event, what we learn actually and what we train on is the actual time sequence in terms of the probability curve. So supervised training on those data sets is tricky because out of a large number of people, you only have maybe 0.4% who actually get over their lifetime but get cancer. Once you have a method, get that particular cancer, pancreatic cancer. Once you have a method trained, then you can go to prediction, and you can apply this now to a very large number of people quite inexpensively. You use the model to predict whether or not somebody is getting cancer, and you have to pick an operational threshold. People above that are at high risk, below that they are not, even though you can also work in terms of a gradation of risk. And then the challenge is to then proceed with those at high risk and do clinical screening. We pick a fairly small number at high risk. Why? Because for something like pancreatic cancer, the detection methods are not very well developed, and so only expensive tests like MRIs, endoscopic ultrasound, and so on are available. So you want to start with a small number to test out these methods until eventually you can get to population-wide screening when prediction gets better and when the testing methods become less expensive. So contrary to mammograms, initially we focus on which is possible in a large population, we focus on those at highest risk. So technically we ask some questions that what methods works best? Do we have a method that will transfer from Denmark to the US, for example? And what would the scope of a surveillance program be, as I just mentioned? So technically, transformer is the best one of several methods. It's harder to make a longer-term prediction whether somebody gets cancer within 3 years. It's hard to predict whether somebody gets cancer within 12 months. What do you use as input? Disease codes. We now have new work that I'm not showing right here. It's about to be submitted. Adding medications, actually, in addition to disease codes, which are standardized internationally as our medication. So more and more data is better than less data, obviously. And then, of course, it is harder to explain, to predict whether or not cancer occurs in a larger time interval. So those are some of the technical questions. And we focus then, typical report is area under the curve, what it might be, and you can see, 1.0 is perfect, and this is random. So you want to be up in the upper left-hand corner. You know these curves, true positive against false positive rates. And that's interesting if you want to compare different methods. But if you want to apply something in real life and make a prediction, you have to pick a decision parameter somewhere along this curve. So we focus on the left area here, which is low recall. Why? Because we want to reduce false positive rates. That in the initial stages, because as you roll this out into the clinic and convincing clinicians and working with clinicians to do this, you want to make sure that actually the false positive rate is low and the positive predictive value, PPV, is as high as possible. So there's an art of how to get that right in the usual confusion matrix that you're familiar with, and you optimize the decision point that you use in real life. The best way to report this, I think, is actually to think of a large number of patients, say 1 million patients, and then along this curve, you take smaller and smaller and say take 1,000 out of 1 million, and then you can ask, how good is the prediction relative to what you would do if you just take into account the incidence in the population without extra information, maybe stratified by gender and age. So we are about a factor of 30 or 70 better, depending on the healthcare system, and this also reflects the quality of the data. And there's ongoing work. We've now proved it by adding medications, and one also would like to add blood values and so on. There's technical work to be done in the future, and this, of course, calls for collaboration. One can assess whether or not somebody is at high risk, whether or not they're likely to actually get the cancer, within a short time or a longer time. And you can see that if you look from the 3-year period, actually there's a decrease in information content, and this is important to analyze by about a factor of 4. So it's very important in these prediction methods and application, and to develop trust. It also has to do with interpretation, to make sure to assess where is the information content in this case in the clinical records, and where is the information that's in terms of actually being able to predict the time interval at which the cancer occurs. Then of course coming to interpretation of another kind, to look at features. As you know, this is complicated. You use something like IG gradient or something called the Shapley method. It is not easy because out of 2,000 disease codes we use, only a small number actually have a high predictive value, and some of them are quasi-symptoms like unspecified jaundice, or of course if somebody has pain and so on. The art is you're training on 2,000 disease codes, and then you report using one of these methods the isolated contribution of individual factors, and that's technically actually quite challenging because machine learning does things in context, while this is picking one out at a time, and each one alone does not in an additive way together with others then predict the occurrence of cancer. So that's challenging, and that's of course ongoing technical work in the field. We have some data also in terms of predictive value for black and white, and male and female. It turns out for white persons in the U.S. Veterans Administration system, that their predictive value is higher. For females, slightly higher than for males. You would learn the bias, look up the bias that the machine learning method has derived from data sets which are uneven, mostly because of socioeconomic differences. And so to then do that better in the future, once you know where the differences are, where the potential biases, you have to actually do several things. Either you change, either you train separately on different subgroups, say black and white, if it's ethnic or socioeconomic, and or to change the machine learning method not to learn those distinguishing features that are not predictive of cancer, but actually take those out and get a method that will predict with even accuracy across different groups and reflect the difference of occurrence of cancer in those different groups. So that's a technical challenge, so that we are currently working on, and are others. You can look this up in a publication in this journal here. Also, there was a news clip on CBS News that actually became generated some public interest in the US this one year, PDAC risk-CBS. And importantly, for us, and I think maybe for this kind of effort, we make this software available in an open source fashion so we can generate collaborations. Lots of challenges in this, which I can't go through right here, but most importantly, it's important to remove the bias that's inherent in these methods because of uneven databases and, of course, uneven healthcare systems, and then to actually access to the right kind of data sets and computing resources, which is quite non-trivial. Done very well in Denmark and the Vegetable Administration. Others are in the works in Germany, for example. In the UK, I talked to Tim Hubbard about that some time ago. UK Biobank, we already have access. Others, that's going to be a major opportunity because the UK has one of the best organized healthcare systems. The other challenge is to accelerate this in a way that gets to the goalpost in a small number of years rather than a longer time. And so I plan to collaborate with others by using a network effect, perhaps using one of the grand challenges from Cancer Research UK, the other organizations. And the important thing is to collaborate with your friends or collaborate with people who are highly open to sharing. And I think that's a major challenge of how to get there. You may find it interesting to look at this book, Early Detection, that just came out by a philanthropist who talks not only about the scientific problem, but also about the economic and social barriers one needs to lower in order to be able to get to early detection of cancer. If it's successful, of course, that will be of major benefit, we hope, to patients, and also it should bring down the cost of cancer care. People will die, but they'll die of something else. But cancer treatment is very expensive and wants to bring down the cost of that. And that's an important aspect. So overall, we want to build an alliance that, having worked on this step one that I just reported on, ongoing work, to work with others who do sensitive detection methods, such as mass spectrometry or antibody-based methods, as tested recently in the UK Biobank cohort. Very exciting new developments to be established, but that's also absolutely essential. Once you have a high-risk population, you want to be able to detect the cancer in those. Some false positives, yes, but with a reasonable subset of those patients, you want to be able to actually detect the cancer as early as possible, and then either have early therapy or other kinds of intervention. So I hope to build an alliance going beyond this particular collaboration, and I'm very interested to talk with others. I talked to the Early Cancer Institute in Cambridge, which is a very interesting organization. You may know about them, and that's it. Thank you very much.
Awesome. Thank you so much, Chris. So we are open for questions, maybe one or two questions from the audience. Priyanka? Yeah. Maybe while she's bringing the mic, I have one question, Chris. I think these are like very interesting in like if you aggregate it, as you said, across multiple centers and then you keep track of patients. I think that would be kind of like a really good data to feed into like large language models and take the text representation from them. But then this type of data is also notoriously dirty in that sense. That like a lot of missing values. Not everywhere is like not all the data sets are like UK Biobank. So they're like a lot of missing values. And then, so how do you think about like what do you think about like handling these missing values and then feeding them into the modeling and like or like the domain shift and domain effects that might be stemmed from these differences?
Yeah, that's a good question. We see huge differences between different healthcare systems with the accuracy of the data. I could go on and tell you detail that's just really quite striking, where even the cancer diagnoses are inaccurate. You have to have a cancer registry to look at that. Missing values, yes, there's quite a bit of that. Time distribution is uneven. A main thing to do is to use the different types of information jointly. Mm-hmm. So, for example, if you have disease codes and somebody gets a diagnosis and maybe that's inaccurate, you may want to look at the actual records after the diagnosis of cancer and look whether or not the treatment they got and the procedures they got are consistent with the diagnosis. We also are starting to do, as you just mentioned, natural language processing. It doesn't have to be very complicated in terms of large language models. Mm-hmm. If it is large language models, they should be specific to the domain. Mm-hmm. But fairly simple extraction of the relevant features from medical notes is one way to do it, and that's work in progress. The important thing is to capture in a clinical setting the type of information that's more reliable because of medical ethics. Clinicians will typically maybe enter the wrong billing code, in the US in particular, different in the NHS, but they will not necessarily prescribe, and they won't do unnecessary surgery. So together, these different data types, you can develop some methodology that extracts the most reliable data items. And in the same way, if you now interpret those features and you want to work with clinicians, convince clinicians that this is something that's worth doing, then the interpretation matters. And even though interpretation is technically difficult of the contributing features, it's important to develop trust among clinicians. And for example, pancreatic cancer, they know that diabetes type 2 and obesity and unexpected weight loss are quite predictive. So you want to convince them that machine learning takes that into account, and we can do that partly with the best possible interpretation methods.
Awesome, thank you so much. And any question from the audience?
Okay. So you mentioned some of the challenges, and the countries that face the data infrastructure challenge are also the ones that get hit by the socioeconomic and racial bias. Do you have any thoughts of how we can work towards overcoming those issues?
Yeah, I mentioned that briefly, and I went to a conference where this was the major topic of a whole conference. This was in the US, and there's a lot of interesting work going on. The difficulty is that the typical studies are epidemiological on relatively small groups of patients. So we're now testing this methodology on millions of patients to look at those differences. How do you address it? As I mentioned, one can put into the machine learning, this is my favorite solution, terms into the loss function actually says do not predict the difference between black and white or male and female, but predict only the cancer occurrence. And it's possible to do that, we think. It's a work in progress. Either that or you train separately, and then you do it in a way that actually evens out the bias in the prediction system. And as I said, it's important, though, to make sure that you capture the actual real-world differences. For example, higher occurrence of pancreatic cancer in black versus white and other cancer types. Black, white in the US is important, it's mostly socioeconomic. So you have to capture that realistically, at the same time for the AI not to predict whether or not somebody is white or black. And it's a subtle difference. But I think it's technically possible, and I think that's the best way to do it.
Awesome.
Thank you.
I have one more question based on what you said. So there will be actually like a lot of challenges with the biases, right, for these models. They might be, so I think debiasing would be another issue here. Yeah, I think cool direction. Yeah.
I mean, there's, as I said, there's bias in the healthcare systems, obviously. And it's more striking in the US, I think, than in other countries. But, so there are real differences in the healthcare practices and application and prospects and so on. And there are also biases in the prediction methods, as we heard, like the images analysis. So one has to separate the two, remove the bias that's inherent in the AI methods because it trains on uneven data sets and get to a point where that's removed, but the real differences remain.
Awesome. Thank you so much, Chris.
Okay, thank you.
Yeah, have a nice day.
I wish I could be there another time.
Yes. Great. So we'll move to the next talk. So we have Mariana Boroni from Brazilian National Cancer Institute, INCA. So Dr. Boroni is the head of bioinformatics and computational, bioinformatics and computational biology lab at the Institute of National Cancer.
National Cancer Institute.
Yeah. And then her group is interested in data analysis and development of workflows for immuno-oncology interface with a focus on high-throughput sequencing approaches for cellular and spatial resolution. And she's also co-founder and bioinformatics leads in OneSkin. That's cool. Okay, awesome.
So good afternoon, everybody. It is it's a pleasure to be here. I really would like to thank the organizers for the invitation, in special Priyanka. And I'm going to talk a little bit about what we are doing in my lab in Brazil. So I would like to start saying what a nice moment to talk about AI in biology, right? Since our community has recently recognized the importance of AI in biology, and I think it's very nice. And for people who are transitioning from biology field to computer science, I thought it would be helpful to start defining some concepts. For instance, what is AI, which is a very broad term that kind of assume it's related to algorithms that are trained to perform tasks that are normally associated with human intelligence. And this concept is also related with other computer science fields, and it represents different aspects and levels of complexity, and I'm not gonna, of course, define all these concepts, but when it comes to biology, I think the community has traditionally used lots of machine learning algorithms, which sometimes are very easy and easy to explain the results, the data. And our lab is mainly using very traditional methods such as regression and clustering methods, even though we recognize how deep learning has shaped the biology field recently. So one example, for instance, was the development of this algorithm where we were able to correlate epigenetic markers with chronological age in order to understand what would be the epigenetic changes that occur in our skin while we are aging. And this was nice not only to understand which are the markers, like the biology concepts behind aging in the skin, but it also turned to be a very nice tool to measure the efficacy of treatment when it comes to anti-aging products. We also applied some machine learning algorithms to develop a tool to diagnose a specific type of acute leukemia with this KMT2A rearrangement, which mainly is detected with DNA sequence, but we were able to show that even though using RNA-seq expression data, we were also able to detect the rearrangement in a very nice way. But what I would like to talk today, it's an ongoing project on our lab where we are developing this prognosis predictor for ovarian cancer patients. And just to give you a context behind, ovarian cancer is it's very incident, not only in Brazilians, but worldwide. It occupies the 7th position in terms of incidence. But when it comes to mortality, it is considered the most deadly, like the most mortal gynecological disease. And the subtype that's more prevalent is this epithelial subtype called high-grade serous carcinoma, which comprise 70% of all diagnosed ovarian cancers. And this subtype is related with some known mutations such as BRCA1, BRCA2, also TP53, and is characterized by a very poor prognosis. And one of the reasons why we have this poor prognosis associated with high-grade serous ovarian cancer is because it is a disease that's very difficult to detect. Most of the symptoms related to the disease are symptoms that are common for women, such as bleeding or fatigue or loss of appetite, so they are generally confounded. And this makes, as I said, difficult to diagnose the disease, and most of the tumors, they are diagnosed in advanced stages. And when it occurs, we have a very significant drop related to survival rates. So the chances of responding to chemotherapy in late stages really drop in those patients. So we thought that maybe we could develop a prognosis predictor that could help clinicians to stratify patients and also to do a better clinical management of them. So our idea was to start working with the Cancer Genome Atlas dataset from ovarian cancer samples. So we downloaded this data, and we splitted the patients in two groups. We called those who had a survival greater than 3 years as good prognosis group, and those who had a survival less than 3 years, we call them poor prognosis group. And we decided to work mainly with microRNAs for this task because we know how important microRNAs are in terms of key gene regulators, and also because they are usually easy to detect not only in tumor samples but also in liquid biopsies. So we thought maybe if we want to translate this technology, microRNAs would be a great shot. But then we have this problem regarding dimensionality because in our data set we have around 300 samples, but more than 1,000 features. So in order to apply machine learning algorithms, it's very important to do the feature selection step. And for this, we created this pipeline where we selected for microRNAs that were highly expressed in the samples. We filtered out microRNAs that were highly correlated, and also those with low variability. And we applied 3 different algorithms. They all perform different regressions to kind of select microRNAs that were associated with overall survival. We also performed this task in your right, to detect microRNAs that were differentially expressed in tumors in related to normal samples. And for this, we use different data sets to obtain these differentially expressed genes. So in the end, we selected 47 microRNAs. So microRNAs that were selected at least for 2 of the different methodologies that we were using for the regressions, and microRNAs that were at least detected as differentially expressed in 2 data sets. So the next step was to train the model, and for this we tried different algorithms, but the one that showed the best performance was random forest. And again, because our data set was not that huge, we decided to do cross-validation. So to adjust the IP parameters, and we also had to do the sample balance using smooth. And to have a hint about the importance of each microRNA, we used SHAP values to understand how the model was ranking each feature. As I'm showing here, we trained not only one model with all the 47 microRNAs, but we thought, well, maybe if we want to translate this algorithm, it would be nice if the model could also perform with less microRNAs, because then to go to clinic, this would be easier. And for our surprise, when we trained with only 11 microRNAs, like a subset of the 47 that we initially identified, the model was still performing very good in comparison with the other results. So as I'm showing here, this 11 microRNA-based model had a great performance in around 0.8 of accuracy. And as I'm showing here in this Kaplan-Meier curve, when we split patients, like the test, when I'm using the test data set and split patients according to their prediction, we can see that the model was able to split patients between these two groups that reflect their overall survival. And what was interesting is that none of the microRNAs that were selected, even though some of them, they were already, like they could be already used as biomarkers because alone they are associated with patients' prognosis. But when we compare the accuracy of each individual microRNA, none of them had a higher accuracy than our model combining all these microRNA signatures. We also compared this microRNA signature with other features that are normally used to evaluate ovarian cancer patients' prognosis, such as stage, aging, molecular subtype, and we saw that this panel of 11 microRNAs was completely independent of the other variables. Next, we try to implement also these other variables such as age and MUC16, which is XAIB24. It is a serological protein that is also currently used for prognosis in clinics. And we were able to improve a little bit the accuracy of the model. And here, as you can see, age is one of the most important features for this prediction. And we also evaluated each microRNA, how it was related to cancer prognosis. Here I'm not disclosing the name of the microRNAs because we are in an IP process. But for instance, for this microRNA 4, we evaluated all the targets of this microRNA, and we saw that those genes that are modulated by these microRNAs, they are involved in pathways such as apoptosis, tight junction interactions, which are all processes well described for tumor progression. And also using single cell data, we saw that most of the targets of this microRNA is expressed by malignant cells, showing also the involvement of this microRNA in the tumor biology. We also saw in literature that this microRNA have been already associated with proliferation, migration, invasion, apoptosis in ovarian cancer. And it has also been shown that this microRNA is more expressed in metastatic samples, which it's totally related to our results. So showing that we could kind of trust in the model. So next, we thought, well, let's see if we can implement this in clinics. And for this, we selected some patients from our tumor bank. And we designed primers for each of these microRNAs, and we measured their expression using qPCR. It's important to mention that we trained the model with RNA-seq data. So now we were trying to see if we change the technology, if the model was able to predict the prognosis of the patients either way. And for our surprise, the model showed a nice accuracy. I know that this is a very small data set of patients. We are going to enhance this number. It's our next step, but we were happy with this preliminary result. So with that, I think we kind of show the proof of concept that those biomarkers, they could be applied in the clinic, and they could help clinicians to empower them to take a decision whether they and how they should treat those ovarian cancer patients. But one thing that I would like to comment is that although in the last years we are seeing an enhancement in terms of publications and studies to identify biomarkers, what we are seeing in terms of approval of these biomarkers is it's a different tendency, right? And one of the reasons why I believe this is happening is because we as scientists sometimes we tend to look for complex problems as like simple problems and sometimes focus on just one layer of this information. Maybe it's not enough to have the whole picture. So that's why we also want to develop new methods thinking about how we can molecularly profile those patients, but now considering multi-omic layers to have a broader view of this problem. And I think the way we are moving towards is not combining only different omics data, but also different data modalities. And I think this also comes with lots of opportunities for us to apply this in clinics and not only to improve treatments, but also to prevent disease, right? I think there are lots of challenges in implementing this kind of methods in healthcare. Some of them we have already discussed. I want to emphasize maybe data quality and diversity and how the data sets that are out there, they really represent us in a general term. And thinking on that, we are now starting another project in Brazil, and our group is especially focused on gynecological cancer. So we are going to profile multi-omics data from 3 different cohorts of ovarian cancer, cervical cancer, and endometrial cancer. And the idea is now to have also Brazilians profiled, because we are kind of underrepresented in data banks that are available right now. And I think with that, maybe we can identify markers actionable biomarkers that can be, more adjusted for our population. So with that, I would like to acknowledge all my group, in special Cristiane, who performed all the computer analysis that I was showing today. Also my collaboration, Professor Alexandre Quevedo from USP, and the funding agencies. Thank you all.
Thank you. Are there any questions?
Can I ask one?
Yes.
So you mentioned something about random forest model that you're working on. So one of the problems that I was wondering when you are using random forest model is a lot of trees can cause computation problems in your algorithm and can lead to ineffective predictions sometimes. So have you ever faced such kind of issues with your prediction model when you were testing your model or something like that?
Yeah, I see your point. And actually, we have tried different models, but random forest was the one who performed the best. But one thing that I really would like to do is to do embedding, and where we can do different models and maybe a votation in the end, because then I could avoid this bias from one specific model. But you are right. But as I said, we tested several different models, and this was the one who performed the best. Thank you.
Yeah, thank you.
Any more questions? No? Okay, so I guess that's it. Thank you so much.
Thank you.
Okay, our next presenter is Dr. Stein Aerts. Stein is the director of VIB Center for AI and Computational Biology. He's also a group leader there and professor in KU Leuven, Department of Human Genetics. His lab has been pioneering and developing machine learning and also computational tools for analysis of epigenetics data. and it's a very diverse and multidisciplinary portfolio of research, combination of single-cell machine learning and high-throughput experimental approaches. We're happy to have you here, Stein. Yeah, here you are.
Thank you so much, Mo. Thanks, everybody, for the opportunity to be here. It's really a pleasure to be in this conference and to present our work. So my lab is interested in deciphering the genomic regulatory code that underlies the spatiotemporal control of gene expression, all the genes in our genome ideally, and then all the cell types in an organism. And with the spectacular increase of resolution and scale of single cell and spatial technologies, as here you can see some example in the whole fruit fly, in the mouse brain, in an orchid flower, in zebrafish development, we can start to dream, let's say, or envision having AI models that can generate such maps from the genome sequence. And ideally, from these models, we hope to explain how the cis regulatory system, the regulatory systems work, where are the control sequences, enhancers and promoters, which transcription factors bind and recognize these, what is their cooperativity, who is activating, who is suppressing, how do multiple regulatory regions, sometimes very distally, come together and determine the, let's say, transcriptional rate of every gene in the whole genome. And such models would, and of course, be very powerful to assess the impact of genetic variation, because we could change one nucleotide and pass it back through the model, and would also boost synthetic biology applications. Now, this image is, of course, a bit in the future, and it's interesting to think about, but we're already progressing as a field quite well, I think, particularly with models that are trained, let's say, locally on epigenomic tracks. That can be, for example, chromatin accessibility, DHS block attack, or transcription factor binding, some examples here from literature, enhancer activity data, and we've been mostly focusing on training such models on single cell attack data. So these models learn how to predict from the sequence of an attack peak, let's say, in which cell type or cell state this region is accessible, and also the height potentially with regression models. There are also models that take larger receptive fields into account, and that can now also better and better predict gene expression, such as these examples here. And also with DNA language models, there's also more and more progress to find insight into this regulatory system. Because indeed, we don't want to train these models to predict chromatin accessibility, because we can measure that, but to explain at nucleotide resolution level, ideally, why this prediction is made in a certain way. For example, here you see nucleotide attribution scores. I will show several examples in the talk that we can link to transcription factor binding sites, or challenge the model with different input sequences, mutated sequences, and assess where it would affect the prediction, and then compare that with experimental data. In this case, that you see it agrees very well between the predictions and the experiments. So as a first example, I'll give you, go through the model that we trained on the fly brain. We generated single cell attack data of the whole brain, and for every cell type here in the rows, you have attack peaks, which are very specific for each cell type, but also regions that are accessible in in combinations of cell types, and so on. And then we basically start from the raw single cell attack fragments and perform first topic modeling. This optimizes for each topic, this latent variable, let's say, two probability distributions, one over the cells and one over the regions. So the one over the cells we use to cluster the cells. This is the dimensionality reduction. And the one over the regions we can use to label the regions, and then train a classifier on that. So this is an example here where topics can represent specific cell types like Kenyan cells, or all glia, or all neurons, and so on. And then the model is trained to predict that from the sequences. Then after it's properly trained, we can investigate highly predicted sequences by the model and get these attribution scores and assign them to candidate transcription factors, because often these high important nucleotides come in blocks which represent transcription factor binding sites. We are in the fly, so we can take these highly predicted sequences and test them by cloning them with a minimal promoter GFP and making a transgenic fly, and then we can see indeed that this enhancer predicted for T4 neurons is very specifically active in T neurons, and this one predicted for Kenyan cells is very specifically active in Kenyan cells in the mushroom body of the fly brain. We did this for many enhancers with a very high success rate. This is published. and we can find many genomic enhancers that are specifically active in Kenyan cells, as you can see here as these examples. Running this over all the predicted sequences in the genome allows us to collect all these seclets, cluster them with TF-Modisco, and generate what we call code tables, where for every cell type you see here a combination of often used patterns in a certain combination. And we can link them to transcription factor candidates by their expression, because we also have single-cell RNA-seq data on the same system. For example, this fork-head motif, this pattern is used by the model to predict accessibility in these T neuron subtypes, neuronal types, and that's exactly where fork-head is expressed, but it's not expressed in T1 and T2, and that's also where the model does not use its pattern to predict accessibility. And that's also true for ACJ6 and for TFAB2 and so forth. Interestingly, the model also uses patterns negatively, so when this pattern is present for the MAMO binding site where MAMO is expressed, the model uses it to predict that the region is not accessible. A third way of explanations that we can do with this model is by saturation mutagenesis. So we mutate every nucleotide into every other nucleotide, score all of these with the model, and find positions that are heavily affecting the prediction score. We also made these mutations, made neutron-genic flies, and indeed this is the wild type, completely abolishes the activity of this enhancer. Or predictions where the enhancer is predicted to be higher because it destroys these repressor motifs, and also there we found an increase of the activity. And then a fourth, I actually put this here under a fourth way of explaining enhancers, because we're using synthetic design also as a way to get insight into enhancer sequences using these models as kind of oracles. So Ibrahim designed or came up with 3 strategies to do synthetic enhancer design. One is generative, a GAN, in fact, and the other 2 are, let's say, more white box state space searches, where you start from a random sequence and find your way through a local optimum. We use the greedy search here, but you can use a variety of search space algorithms. So in the first example, we start with a random sequence, mutate every nucleotide, select the mutation that the model gives the highest delta increase of prediction score towards a target class, and then repeat this process until we have a high prediction score. We can do that over thousands of random sequences to begin with, and then have a population, and you see that almost, well, none of them, random sequences, have a high prediction score in the beginning, but as we make more and more mutations, after 10 mutations, we already arrive at very high prediction scores, as high as in the genome. And then these look like this. So a random sequence would often have a very low score and would have these repressor motifs present, while after 10 mutations, you see that these repressor motifs are gone and replaced by activator motifs. And in this case, for Kenyan cells, we find exactly those motifs back that we have in the code table of the genomic enhancers. So this comes more or less to the centroid of the enhancer of that cell type. We can again test these in the fly, and we found indeed that they are specific, selectively active in the Kenyan cells. So this seems to work, and some did not work from the start. After 10 mutations, we gave them another 5 mutations, and then they worked. So we can make almost all of them work, and some of them are very, very specific and very strong in the Kenyan cells, in the fly brain. We repeated this whole process for a second cell type, but now starting from the same random sequences. So this is interesting because, again, we can evolve them according to the model to another class. Now, pyramidal glial cells, completely different cell type. Here again, after 50 mutations we now took, we see very beautiful pyramidal glial motifs that correspond to the combination of motifs found in the genome according to the model. And in this case, also the enhancers worked in pyramidal glial cells, showing that the intervening sequences that the model here by attribution scores does not deem important, neither in Kenyan cells nor here are likely not contributing to this enhancer activity. A second strategy that is quite similar, but now not by point mutations, but by inserting motifs that we already get from the code table. We can put these motifs at every position of a random sequence and select the one with the highest prediction score and then insert a second motif and so on. Doing this over thousands of sequences gives us distributions where we can see that the combination of these 2 motifs, MEF2 and ILS, gives a very high prediction score, and we can also investigate where they are located next to each other, what the models finds the best locations, finding 5 base pairs and 3 base pairs between the motifs as optimal spaces. That also led to some of these synthetic enhancers that we generated, where the 3 motifs for MEF2, ILS, and 1-cut are very close together, And we also decided to cut out that sequence, and it actually also worked. So this is a way to design minimal enhancers of, in this case, only 41 nucleotides that drives the same expression pattern as the 500 base pair enhancer. So enhancers can be made only with motifs in this system at least. Coming back to these repressor motifs, they really work. So we did an experiment where we took this minimal enhancer that is beautifully active in Kenyan cells and asked the model to evolve it to not working anymore. So can the model choose mutations that destroys this enhancer without touching the 3 activator motifs? So these nucleotides are protected, and it can do that. It needs only 5 mutations to do that, 1, 2, 3, 4, 5, and each of them generates these repressor motifs. And now if we clone that enhancer and make a transgenic fly, it is no longer active. So in this case, we can really see that these repressor motifs, so if you have enough of them, can actually dominate over the activators, because the activators are present in the cell, the transcription factors are present, the motifs are there, but it's not activated. We went on with some more elaborate experiments also to use enhancers that are active and accessible in 2 cell types and evolve it to destroy one of them and to keep the other one. This has worked. And also to add cell types to one enhancer. So this one is active in the optic lobe here, and we can evolve it to keep that and to add a second cell type here, the Kenyan cells, and now it becomes also active in the Kenyan cells. So you can do a lot of interesting things here. A new thing we have made now is a software package called Crested. There's no preprint yet. We are still working on that. We still need a bit of time, but we already put it on GitHub. And the Crested Python package, you can do all the analysis from the raw single-cell attack data in the SCVerse framework. Then choose different architectures and different ways of regression or classification to train the model and to follow up with the insights into the logic and to do the enhancer design. And we called it after the great crested newt, which is an endemic species in Belgium. That's a salamander with attack peaks on its back. So that's why we like this name. We also took this package and models to a further test in an open challenge organized by the Allen Institute of Brain Science. And in fact, the crested models had won the challenge. I won't talk about it here, but you can look at the preprint. Niklas from the lab has participated a lot in that. By the way, of course, as you can understand, this is also interesting for applications, because if we could make an optimal enhancer for a specific cell type and clone it with a transgene, package it, and deliver it, it could help in making gene therapy more specific and safe. And we actually have a project ongoing in the lab where we're optimizing the pipeline, starting from single cell data, modeling, crested models, design, multiplex parallel reporter assays, and in vivo assays, to be selecting very specific enhancers potentially of interest for gene therapy. This crested package and the models are now also more and more scalable. So Niklas managed to train models on the full mouse brain with 2.3 million cells. And you see here in the to compare the correlation between the actual height of the attack peaks and the predicted height, which correlates well, that this happens to be the case for the majority of the cell types here on the diagonal, and also off diagonal, where cell types are similar to each other in attack, also the model nicely finds this back. So these models seem to work well. He also trained a model on the full human brain, and now we can start, because human and mouse cortex, or human and mouse brain in general, have a lot of orthologous cell types, and so we can use the mouse model to score human sequences and the human model to score mouse sequences as well. Here's an example of that. In the CSF1R intron, there's a very famous enhancer called the FIRE enhancer, which is has an attack peak only in microglia. And this is an experimentally validated enhancer. We can ask the mouse model what are the important nucleotides, and it will give us these, which actually agree very well with experimentally validated transcription factor binding sites. And here the human model finds a nearly identical explanation, which has been trained on completely different data, in fact also on a different genome, and still agrees with this explanation of this mouse enhancer. This confirms and is encouraging for what the model knows, but also can be taken a little bit further and could lead to conclusions that in fact in microglia, human and mouse microglia likely have a conserved trans environment, because it's of course the transcription factors that interpret this enhancer. And if they interpret this in the same way, which we can approximate by the model, we could reason that they are conserved. That principle we could take a little bit further. We tested this in a more difficult scenario, let's say, with more evolutionary distant examples. Using the brain of a bird, chicken, we performed single-cell multi-omics on the chicken brain, finding many of the conserved cell types back, like microglia and astrocytes and interneurons, but also a bunch of glutamatergic cell types which are not really known, and whether they would be similar or not to the glutamatergic cell types in the layered cortex, which is mammalian specific, was not known. We performed spatial transcriptomics with StereoSeq, but also with NovaST. This is our in-house custom spatial transcriptomics platform, and managed to map these unknown glutamatergic cell types to specific locations in the chicken brain. So now we can take all these cell types, take the attack peaks, because it's multi-ohm data for each cell type, and score the sequences of the chicken attack peaks with the mouse model, and ask the mouse model what cell type would this be accessible in the mouse brain. Second approach, we can actually look at the attribution scores and calculate the correlation between the attribution score of a certain class in chicken with all the classes in mouse to see which one correlates best. So you see here that the chicken astrocyte explanation correlates the highest with the mouse astrocyte correlation, microglia as well. But also for glutamatergic neurons, we find this cluster GLU7 that is most similar to the deep layer neurons in the mouse cortex, and GLU1, which is more similar to the upper layer, so it fell off L23 IT neurons. Thirdly, we can take the patterns across all enhancers and cluster the cell types here, human, mouse, and chicken, according to this pattern or this code table. Here again, the chicken cell types, you see these chickens here, fall inside clusters together with human and mouse, and again, the GLU1 is falling together with upper layer neurons and GLU7 with deep layer neurons. Putting that all together gives us a similarities metric with, using the transcriptome with SumUp and 3 measures of the enhancer code comparisons that lead us to conclude that the non-neuronal and interneuronal cell types are highly conserved in their enhancer logic, but also some of these glutamatergic neurons show similarity. The code tables for the non-neuronal and also for the interneurons are super conserved, so the same combination of patterns here are used in astrocytes with chicken and mouse, and even the transcription factors that we predict based on their expression correlation are also similar between mouse and chicken, which you may think doesn't matter what chicken does, but it gives us a lot of confidence of the predictions that we get on mouse and on human, of course. Well, for one enhancer, we found similarity of GLU1 with layer 2-3 IT in mouse, and actually cloned this chicken sequence, made AAV particles with GFP, and we find indeed, although it's a little bit noisy here, but the majority of the signal is indeed in the upper layer of the mouse cortex. So this chicken enhancer of some unknown glutamatergic cell type seems to still work, or seems to work in the mouse upper layer neurons. Another way of getting to explanations, in this case of genetic variation, again using these crested models trained on single cell attack, this is an ongoing work. It's quite preliminary, but I think it's interesting to think about. We finalized a large data collection on 190 human brain donors for 2 brain regions, cingulate cortex and substantia nigra, generated for millions of cells, single-cell RNA-seq and single-cell attack-seq data, trained crested models using this large data set, and also sequenced for all these 190 donors the whole genome with long-read sequencing. So we have beautiful diploid genomes with all the SNPs, indels, and structural variations, and all the haplotypes. And now we can score these variants with these models. And this is an example of a variant that reference G has a high prediction for oligodendrocyte enhancer, and the alternative A here, one SNP, has lost the prediction score for oligodendrocyte. It's indeed an oligodendrocyte-specific peak. And we haven't used the variation across the individuals yet, so now we can look at the chromatin accessibility data of all the donors. So all the homozygous reference Gs here have a high accessibility at that position, and the homozygous alternatives have no accessibility at all, as the model predicts, while the heterozygous here are in between. And all of those reads, nearly all of them, there's a few not, nearly all of them of these heterozygous individuals are mapping to the reference allele, showing a very strong allelic imbalance here. So this is a way to validate the predicted variants that affect enhancer activity using CA-QTL, chromatin accessibility QTL data, and we find hundreds of variants where the model says high increase, it's a high increase, high decrease in activity, it's a high decrease in accessibility across this cohort. I'm okay with time? Yeah. The final thing is a bit more difficult. We try to go further to explain from these models also gene regulatory networks. So these crested models, they don't give you, that I explained so far, they have no insight into gene expression, no gene regulatory network, but also Borzoi and Former, say, Scooby, Decima. They predict gene expression at the bulk, although for Decima now at the single cell level, but they don't provide us with a gene regulatory network, who is binding where. DNA language models also not. So we're trying to get the GRN out of this. Gabriele is using all these models, types of models as a base, and then fine-tunes it using single-cell multi-ohm data to a more white box model. It's a combination of deep learning architecture, but it has some white box, let's say, parameters, namely a matrix of all the transcription factor weights to all the regions where they bind, and a weight for all the regions to which gene they may regulate. And so by this additive linear combination of transcription factor importances per enhancer and of region importances per gene, we try to predict the gene expression and the chromatin accessibility of every cell using the TF expression as input. And this model still has the capacity to go to the sequence, also the capacity to do perturbation predictions, because we can put a TF to 0 and pass it through the model. But it is also open because we can look which TF binds to which region according to the model. It's called Deep Scenic. I don't have time to go over all the validations. This will be in a preprint soon. But we can predict gene expression, chromatin accessibility, and also TF binding to regions, as well as region to gene accessibility, and the most important TFs per cell type. I think Mo is alluding me to stop almost. So, and this seems to work. Just for the last few seconds, I wanted to briefly introduce our new center, VIB.AI, which you alluded to in the introduction. So we are setting this up in Belgium as a 10th VIB center. It's focused on AI in biology. And we're, of course, taking advantage of all the data, hardware, architectures, machine learning, to go more and more to causation from data and models, but still connected, as I showed also some examples, to the experiment. Minimally to the best predictions, but still very important, and bringing those experiments also in the loop. We are setting this up with 6 labs to begin with around central facilities of machine learning, data core, a shared wet lab where one can outsource their experiments. And we already have my group and our first new group, Joana Pereira. And I just wanted to say all of this because we have 4 group leader positions open. So please consider applying. And these are the affiliated PIs from VIB in other centers that are strong in computational biology, and we form a very new AI-driven community in Flanders, and you are very welcome to explore that through collaborations or even joining. Thank you so much.
Thank you so much.
No.
Amazing talks. Stein's talks always like, you have to keep out like which slide is which method, and then you throw like,
oh man,
yeah, it's like, okay, cool. Do you have any question?
Okay. Hi, very cool talk. I was wondering, do you think that with the transcription factor as input for like this sort of trans-environment prediction, will it be enough to take wild-type cells, or do you need perturbation data? Those, or as in,
yes.
For some transcription factors, it might not vary so much
yes,
across the context.
Exactly.
And second brief question is about the chicken data, whether you find there was a lot consistent, but have you looked at sort of differential attribution between the species to see if there's any sort of fine differences between them?
Yeah, beautiful questions. Thanks a lot. I think for the first question, indeed, you already alluded to that. If there's not enough variation in the data set on which you train this on, for example, states and cell types where transcription factors are high, low, middle, and so on, but everything is flat for a certain TF, then it's impossible to learn that. And if that is a large majority, which is often not the case, but imagine, then I think it's interesting to expand this either across species or across systems, right? Making the universe larger, or indeed expanding it through perturbations. I think that's also very interesting. But we haven't explored this to the full extent, actually. So And then for the second question, yes, that is indeed a little bit of the next step, if you want. So we focused a lot on conservation. It's also easier because, yeah, we get confidence in the model and it's conserved. Everything else, which is different, is either an error, a problem of the model, or evolutionary difference. So that requires a bit more time, and we decided to separate this out. We do have a couple of very interesting observations already, particularly for conserved autologous cell types like astrocyte, same TFs, same enhancer code. Look at the genes, hundreds of genes differentially expressed. So now you actually have the capacity to go and look at which enhancer disappeared or was generated de novo. And we're also working on adding additional species to this, because with the phylogenetic tree, we're going to have much more power to see what is ancestral and what not. Now with 2 species, it's a bit more difficult, but that's indeed the road ahead.
Really cool, thanks.
Thank you so much.
There's some questions online. Would crested also work for highly similar cell type, or does it need a lot of variation? I'm interested in 2 cells. I think it's just 2 specific subtypes of CD8 plus killers and CD4 plus helpers.
Yes, this is what I think computational biologists usually do every day, right? Trying to figure out where's the insight, how can I make it, what's my background model, what's the null model is in bioinformatics. It's the same problem. We do have some models which are trained on very similar cell states and still enable us to find nice differences.
Yeah.
But then it's often also interesting to use a second model where you have a bigger contrast and then combine the 2 models next to each other or cumulatively for the explainability. So I think this is the creativity of the researcher to.
Yeah. I also have a question myself. So regarding the last work, I think you mentioned this work from Decima, from Gökçen, and then also Scooby from Julian, and I think they're like piggybacking on Borzoi and also Informer. They did benchmarking, and then I think the community at some point was like, maybe these models are not working, right? For like they're not working, but basically not capturing really like long range interactions. And now we're trying to predict long range. So this is basically a long range prediction problem, right? When you're talking about cell type specific. So we're basically inheriting all of those problems because we're not fine tuning them. I mean, we're fine tuning, but we don't have enough data for that.
Yes.
So just I think it's very open ended, right?
Yeah.
I just wanted to hear your opinion and comment on like.
Yes.
Is this the right direction, or like, whereas you here have a more mechanistic model, you're trying to screen it?
I agree, and this is what everybody now is trying to understand. So for long-range interactions and for variants that predict changes in expression, this was all not working so well, Informer and Borzoi. We took a bit of a different approach, rather than the transformer that is still used by Decima and Scooby, is that we consider all the regions that we take as input, all the attack peaks, let's say, or the regulatory regions, locally. So every sequence of 2 kb goes through the encoder from that embedding we use to predict TF binding and expression output. And we collect around, in 2 megabases, we collect those interesting regions, and we re-establish the enhancer to gene links by training that, because it didn't work so well in these models.
I see.
So we're trying to overcome that. Does this solve everything? Yeah, of course not.
But this kind of diverges from those like Decima and
it deviates from that because we throw away the attention of the enhancer to gene links because distally it's very difficult, and we collect them all in an array of regions and then see through which sequence embedding and where you are with a prior over the distance also. How can you, which ones do you combine to predict gene expression and train on that? And then see which ones have come out with the highest weight.
I see.
Whether the future and the community are, I think, working well together to find the right way of doing all this.
It's very exciting.
Right?
So
it's going fast now.
Yeah, awesome. And then someone asked whether you have the slides somewhere online or not. I think that the presentation is recorded, so if you can, right, you can watch it. Yeah, awesome.
We'll figure that out.
I think that's it. Thank you so much, Stein. Appreciate it.
You too. Thanks, everyone.
Okay, so our next speaker, please, is Dr. Philip Kim, so from University of Toronto. Philip is a professor at University of Toronto, co-founder and CTO of Fable Therapeutics, and a specialist in machine learning and computational biology. His group combines modern AI methods with high throughput experimental data to enhance drug development and therapeutics, and then he's going to talk about ML methods for protein and peptide design. There you are.
Okay, thank you. Yeah, thanks for the invitation. It's great to be here, and it's great to be at a meeting with so many different talks. So I'll talk about something completely different. I'll talk about protein design and peptide design, and it's great to be exposed to some more biology because I tend to, just look at proteins these days. Okay, as mentioned, so I will mainly talk about my academic work. As the introduction kindly mentioned, I wear many hats, and among others, I run a biotech company. Right. Okay, so this is so the machine learning revolution has really made my life very exciting, and so the last 5, 6, 7 years in protein science have been extremely exciting. And what is the progress has really been tremendous. Has really been astounding, right? So I've been doing this for a fairly long time, right? And after several decades of pretty steady but very incremental progress, we have seen like 5 years of several revolutions happening, just one after the other, and things that were absolutely science fiction just a couple of years ago, we can do that now, right? And pretty much everybody can because most of the software is available, right? So we now live in this time where this dream, and it really was a dream, of making biologics or protein-based therapeutics on a computer is basically reality, right? We're not totally, it's not everything, it's not perfect yet, right? And we're still working towards making it more perfect, right? But it is definitely reality, right? And so, I want to give a bit of a shout out to what made that possible, right? What made it possible is a fairly large body of data that has accumulated over the years, and most notably for my purposes in the PDB, right? So there's hundreds of protein structures in the PDB that form a fairly high quality set of training data, right, that we can use. I want to give a brief shout out to Moore's law, right? So I guess we're going to talk about this now, but I just want to remind everybody that every one of us walks around with a supercomputer in our pocket, right? This used to be a supercomputer, right? Like just when I did my PhD, this was a supercomputer, right? And then of course we'll be at the advent of machine learning methods, and I will do a bit of a walk through what happened in the last 5 years, 6, 7 years, right, in the protein world, right, and I will claim a bit of personal credit, whether we deserve it or not, for some of the advances, and right. So what happened? What had to happen for all these great advances of machine learning to make an impact in protein design was a way for us to encode the data in a way applicable to machine learning, right? And so very early approaches, and I still remember talking about it with colleagues and students about how one would use, and back in the 2015, 2016, right? AlexNet was already a thing. CNNs were all the rage, right? And how do we do CNNs on proteins, right? Protein structure, right? And so the early approaches, and there's papers out there that do that, right? The main problem is you have a protein structure which is a 3D object, right? A protein is fundamentally a 3D object, right? And then CNNs operate in 2D, or like language models like RNNs back then would operate in 1D. And then how do you do with 3D? So the simplistic approach was to use volumetrics, right? So you, instead of pixels, you do voxels, right? And then you make 3D space into little boxes, right? And then you do your CNNs in little boxes. And that never really worked, right? So that's why you never heard about it, right? That never really worked, and we played around with it and never really worked very well. And so one breakthrough came really when people started using graphs, and my group did some of the early work in this graph-based work. So essentially, you can break down your protein into amino acids as nodes, right? And then you encode some information in the nodes. You can encode information in the edges, and that has certain advantages. Like you don't have to discretize, and you get some certain mathematical properties, like you get invariance, right? So it's just nicer for the computer to deal with this in a graph setting. This has evolved now several times, and One evolution was that all the rage used to be equivariants, and then earlier this year AlphaFold3 showed up, and they proved that equivariants, you don't really need equivariants. And if you're interested, then I'm happy to have an in-depth discussion with you on equivariants and why I think it's still a good idea, but anyway, that's a separate topic. So these graph methods really paved the way for a lot of what came after. And we, in our lab, we applied the graph methods, we used the graph methods to develop the first inverse folding, so the first protein sequence design method. So you have a protein structure and you encode it as a graph. You train a model to fill it with amino acids, and then these amino acids will then fold into the protein that you want to. And we make use of the PDB, and we make use of graph convolution, and we could show that this really works at regenerating real proteins. So this was effectively the first method to do fixed backbone design with machine learning. We could show back then, that was the pre-AlphaFold world, but we could still show that all the proteins we design in all alpha, all beta, alpha beta, alpha/beta, they make real proteins. We could make some proteins in the lab, and we could show that they are really folded proteins, right? And back then in the dark ages in 2019, 2018, when we did this was amazing, right? The computer comes up with the protein sequence that actually folds, right? This was like really, right now it's really trivial, but that was kind of an eye-opening moment for me to see that the machine learning in protein design and protein structure really has a role to play, right? And I will note that inverse folding is now considered to be a mature. These approaches now have now matured, right? And the newer approaches, protein MPNN is probably the one that most people use. Is conceptually it's very similar. It's basically the same protein solver, but it is much better engineered, and it is probably the method to use, right? And there's more innovation. There's a bit more innovation happening, but it's largely considered to be a mature problem. Okay, so that's for protein sequences on given a structure. Now we want to design the structure, and so we want to do de novo design, and for that we turn to diffusion. And I'm a little surprised to have had a full day of AI talks, and nobody has mentioned diffusion yet. So I'm going to mention diffusion now. And so people, if you haven't heard about it, I'll give you the 2-second explanation. The 2-second explanation is that you can train an AI and a neural network on learning data from the noise in a stepwise manner. That's not really true, but anyways, that suffices as a quick explanation. And diffusion is an interesting thing that it came out. Diffusion came out in 2015, so like a really long time ago. Nobody really took notice until the first diffusion method started to beat GANs at image generation. That was in 2020. And ever since then, diffusion has been all the rage, and everybody has probably seen DALL-E or Midjourney or Stable Diffusion, all these diffusion-based image generators, but they work really well. So then a new student came to my lab, and he said he's really excited about this diffusion thing, and he wants to build a protein design method using diffusion. And I was like, oh, that sounds like a great idea, but I mean, I told him basically we have to do it tomorrow. And we have to publish it the day after tomorrow because otherwise we're going to get scooped. And so he then went on his way, and we did actually very well. So we were the first validated diffusion model out in the summer of 2022. Before other people like a biotech company Chroma or RF Diffusion or the Baker Lab came out a fair bit after us. So what we did essentially, we took again this graph-based method. And the good thing about a graph-based encoding of the structure is you can actually design it as a matrix, right? And then what we can do then is we use these 60 matrices, right, to get a fairly detailed encoding of the protein backbone structure, right? So you can do distances and angles, right? And we can then train a machine learning method to essentially generate these sort of matrices, right, in a diffusion model, and then you get out the protein backbone, and then you can use a sequence design method like Protein Solver or MPNN or Zeta to then fill it with sequences to get a real protein, right? And amazingly enough, back then it was amazing. Back then that was really like astounding. Amazingly enough, that actually works, right? So you're getting out these sort of proteins, right? And they look like real proteins in sort of any way we can tell computationally, and some of these proteins are really new in the sense that they're very different from anything you would have in the PDB. So they're proteins that nobody's ever seen before, and they really exist. And you can actually make them in the lab, and you made some of them in the lab, and you can show that they're nicely alpha helical, alpha fold sinks, they fold this way, and you can melt them, and they have a native melting curve, like a nice two-stage transition, so they're really well-behaved proteins, right? And so there you have it. So this really works, right? And we can now, and everybody can now, with Protein SGM here, or with Aerts Fusion, or with Chroma, or with Genie, or with there's now multiple protein diffusion models out there that many of them work very well, right? You can now make real proteins, real new proteins just on the computer, right? And that really works, right? And that's just in 2022, this was absolutely mind-blowing, I think. And now it's 2 years later, and it's like completely trivial, right? And I think that's a good gauge of how fast the field has moved, right? That this is true, right? Okay. So now we want to do something useful, right? And as mentioned, I did co-found a company, and I run the machine learning part of this company now, and we make antibodies, right? And we make antibody therapeutics specifically. And so and at Fable, we build I would say a state-of-the-art diffusion model to do structure and sequence design for antibodies, and we can show in these benchmarks here of sequence design and structure design. We outperform the public state-of-the-art by a fairly healthy margin, right? And we can do now antibody candidates that we're now advancing in the therapeutic pipeline. And depending on the problem setting, we can be very accurate, and we can get when we design antibodies and we test them in the lab, depending on the setting, we get hit rates up to 60% or something, right? So as I mentioned, so the dream is very much reality. It's not perfect yet, but it definitely works, right? And there's not only Fable, but there's multiple companies who are doing this. Of course, Fable is the best company, obviously, right? But this is now really happening, right? So the future of biologics discovery, of antibody discovery, is definitely computational and AI driven. Okay, I want to switch gears a slight bit. I'm gonna cover the next two topics very briefly. So I wanna talk a bit about dynamics, right? So that's what I'm academically excited about, right? So we wanna, and that's especially true for peptides or peptide microcycles. So structure design and structure prediction, that's old hat now, right? So that's kind of solved. And so now we want to design, or we want to predict the dynamics, so the conformational energy landscape of a peptide, of a molecule, right? And so those of you who are physics or physical chemistry inclined, and there are some people like in the audience, you may know that there's some conformational energy landscapes that we can show like in this like 2D cartoony fashion, right? And of course, there are methods to sample the conformational landscape using a trajectory-based method like MCMC or molecular dynamics, right? But the main problem is that these often, to go from one state to the other, often take a long time, right? So that's the problem. And so what we want to do, really is we want to get the full energy landscape out in one go. Right, so we want to essentially predict that whole thing. So structural prediction is predicting this one thing here, this one state, but we want to predict the whole landscape, right? And so to do that, we can turn to machine learning, And what we want to do effectively is this is the convolutional energy landscape, right, in a 1D setting. And we won't get a neural network that'll start with a Gaussian and then convert this Gaussian into an energy landscape, right? And that's a neural network. And by the way, that's kind of exactly what diffusion is doing, right? Diffusion converts a Gaussian prior into your target distribution, right? And so we did that essentially. And since we're talking about explainable AI. You know, the part where we're sort of closest to explainable AI is the PEPFlow. This method here is it consists of a hypernetwork and a real network. So this network here actually does the prediction of the conformations, right? And it's a relatively small network of only 100,000 or so parameters, right? But sort of the heavy lifting is done by the hypernetwork, and this hypernetwork predicts the parameters of the smaller network. And then this put together then, and it's altogether a pretty complicated approach, will then generate conformations of all atom conformations of peptides. And so it works really well. So we're getting I would say state-of-the-art results for single structure prediction. But we also get different conformations out, and we're doing quite well at both precision and recall of NMR conformations, right? So of real conformation energy sampling from experiment, right? And as you can see here, right, if you compare to MD, we are of course much faster to sample more conformations. And you can see in MD, you will always have this trajectory-like approach, like all the samples you're getting are similar to each other. With the PEPFlow samples, you're sampling directly from the energy distribution, and you're getting a different minima out right away, right? And so this is of course an advantage. And so now we're using PEPFlow in a binary design manner, right? And the big prices, and I'll skip that. The big prize is to do docking or to do binary design directly. So what we're doing now is PEPFlow++ is using this method to generate bound peptides as binders or then in the future as therapeutics, right? And in the last couple of minutes, I want to do, because we had a couple of great talks about gene regulation, right? So I want to talk a tiny bit about gene regulation, and what we're focusing on is C2H zinc fingers, and C2H zinc fingers are of course are the most common transcription factors in the human proteome, right? So these are C2H zinc fingers. They used to be the main technology people use for gene editing or for all sorts of things, and that because they are highly engineerable, right? That was before CRISPR came around, right? And CRISPR, of course, kind of totally blew them away because everybody can use CRISPR and you can make a guide RNA and you can edit your gene you want, but zinc fingers still have certain advantages, and they're human proteins. So there's many zinc fingers. There's 7,000 zinc fingers in a human proteome. So they're less immunogenic than CRISPR, which is a bacterial protein. They are pretty small, and they can be used to bind any DNA sequence, right? So for instance, especially for in vivo therapeutics, right? So if you want to use it on yourself, if you want to inject yourself with a transcription factor, you probably want a human protein that's non-immunogenic, because your body is not going to like it if you inject yourself with CRISPR, right? And so that's why zinc fingers are still that's why zinc fingers have advantages over CRISPR in this in vivo setting. The main problem is that predicting zinc finger interactions was really difficult. And you couldn't. It was previously impossible, essentially, to generate a code that tells you, given a certain amino acid sequence, what are the given a certain DNA sequence, what are the zinc finger amino acids that will bind it tightly, right? So that DNA to amino acid code was impossible to solve. And that has to do with the complexity of zinc finger DNA interactions. And I won't get into that, but so to make a long story short, my collaborator Marcus Noyes at MOU generated a very large set of data billions of data points on zinc finger DNA interactions, and then we use that with a I would say reasonably smart setup of using a transformer, but a hierarchical transformer to essentially convert DNA targets into zinc finger amino acid sequences that will bind this target. And so this is a classic transformer problem. A transformer goes from space A to point B, right? So the transformer fits very well. And then lo and behold, this works really well, and it blows everything else out of the water what was there before. That's not that difficult because everything out there was really bad, and it works very well at getting you the right sequences. And we tested it in a way to reprogram transcription factors, right? To make transcription factors that can either repress or activate any gene you want to. And here we did it against DPH1, and you can see here. So this is the control, and these are the ones. This is the control, and this is all the things we tested, right? And you can see that every single one of them works at repressing the control DNA, and then 3 of them work really strongly. And we can do it for activation too against a different gene, and you can see here this is the control. We get activation of a background every single time, and some of them we get very strong activation, right? So it really works. And I'll skip the rest. I just want to leave you with this really works, right? We can get single single target specificity, and with a bit of engineering we get very strong and very specific zinc fingers. And I want to close here, almost on time. This is my lab. This is my funders, and as I mentioned, I wear many hats, but some of these are some of that way. Okay, thank you, and I'll take questions if there are any.
Any questions in the room?
Thank you for the talk. That was great. I was wondering if you had any thoughts on how protein language models compare to the graph-based methods, and if you thought that the graphs add an element of interpretability that the language models lack, perhaps.
Yeah. So there is now that sort of philosophical debate around whether we can use tokenization in a language model setting versus continuous representations of the protein structure, right? And so so most of the design methods right now work in a continuous setting with fusion, right? And that is sort of, and that may well just be sort of a inherent bias of the people doing it, because I think that makes more sense, right? And I think many people like me will probably think that makes more sense. But tokenized versions like ESM3 is a tokenization, right? And you can say that it's total overkill because they put in 4,000 structure tokens right in, and then it works really quite well, right? And they get a good accuracy. They come very close to AlphaFold 3, I think, in some of the accuracies, right? So there are some folks in my lab now who are playing around a bit with this tokenization, and we do find, there's been other tokenizations, right? And we do find that ESMC seems to work the best. I think it's still the jury is still out which will win in the end, right? I mean, you can argue, you can make arguments, right, that tokenization at some point, with scaling, you might win, right? But right now, I think that the continuous models are still better. And to interpretability question, right? In the protein structure world, I'm a bit ashamed. I'm a bit ashamed to say this in this Explainable AI conference, but we haven't been overly concerned with interpretability so far, right? We've just been very engineering, like just get it to work, right? I will say though that there is a bit of a debate in the field on on that, but I strongly believe that, for instance, that AlphaFold learns some part of physics, of protein biophysics, right? And sort of part of the metrics you can get on AlphaFold correspond quite nicely, I think, to an energy function, right? So I think that's a debate. Some people would strongly disagree with me, but I'm speaking here, so I can say that. So in that sense, you have interpretability in that sense, right? You don't have interpretability of the actual weighted parameters because there's obviously yeah.
Thank you very much.
Yep.
Okay, so with respect to time, we'll end there and we'll pass the rest of the questions to Philip. Okay, so we're going to close now and come back in 15 minutes after the refreshment break for a panel discussion on responsible AI. Thank you, everyone, and can you please thank the speakers once more?
Right, I think we're ready to go. So thanks everybody for sticking around, and I hope you're looking forward to the end of the day and drinks, and because I certainly am. So for those who don't know me, I'm Richard Milne. I'm an ethicist and social scientist by background, and I actually work just across the road in Wellcome Connected Science, where I lead a team of researchers who work on kind of emerging ethical questions associated with developments in genomics and biodata, which is kind of broadly the framing for this session around responsible AI. And so I'm not going to introduce the panel because you've already been introduced to everybody, but just to say that Mariana made the fatal mistake of mentioning the ethics word in her talk. And I thought, well, and has then very graciously agreed to join the panel. So thank you very much.
Thank you.
And I know, so we did do introductions, but I just wanted to say if there's a kind of chance for anybody on the panel to maybe say anything else about themselves that wasn't captured in the kind of, the little blurb that the various chairs read out. I know, Miha, when you talked this morning, you were talking about that kind of importance in your work of, kind of giving back to patients and your kind of your background. And I suppose I'm kind of interested in starting with this conversation, which is partly about, I think, kind of humanizing AI to maybe humanize the panel a bit beyond affiliations. So, Miha, is there anything that you want to say about how did you get here?
So for us, right, we realized how hard it is to model high-dimensional gene expression data and then to interpret these models like Maria mentioned. I think you did a fantastic job, right, of narrowing down the hypothesis space to 40 or so genes. But when you work with that many features, it's really the interpretation gets really difficult. We learned that the hard way. So what we do today to make our models better interpretable and more understandable, closer to the people, right, and to the doctors we work with, is that we infer biology phenotypes. So in the end, to classify patients in different subtypes or for response, we only work with a handful of features, right? One feature for each biology. And that, I think, makes it a lot easier to explain and to present to the public and just open up the black box that we've built. So to us, it's a lot about feature reduction, and that I think is a big aspect of what we do.
Yeah. And then moving into that, to that kind of interesting that point of explaining to the public, or like who's the audience,
who's the stakeholder.
How about you, Mariana? Who do you see when you're? I mean, I suppose this kind of gets to this idea of explainable AI. When you're thinking about explainability, who do you see yourself explaining to?
Well, first, I think it's important to say that I'm sitting in a hospital in the Brazilian National Cancer Institute, and when we are thinking about those methodologies, especially when applied to the clinics, I think there is, in general, how do I say, they don't give it credit enough. I mean, they are skeptical about those models, right? And to unbox the box, I think it's a way to show like more credibility of what has been done, and also to trust more and to be more comfortable of taking decisions based on what you are seeing. So when we talk to physicians, clinicians about these tools and how they can empower them to take those decisions, they don't understand what you mean by this, right? So if you can somehow show the biology behind, I think it's easiest way to show the value of what you are building. So I think, at least for me, it's very nice to understand also with the machine, like how the biology is behind. I mean, when the machine is selecting those features, it's very nice to understand why they are important, right? What are their role, I mean, in my case, in the tumor progression. And this is also a way to show for people the value of the model per se. So yeah, I can see through these two lenses as well.
Yeah, it's like kind of being in that kind of middle space, I suppose.
Yeah, right.
Yeah. And Philip, so you work in a kind of slightly different context, I suppose. So how do you see that kind of question of explainability in your context? I think you kind of started to touch on it with Ronnie's question at the end there. You wanted to elaborate?
Yeah, maybe we can just elaborate a little bit on that. Right? I mean, so we're in the molecular world, right? Modeling used to be very physics driven, right? And that's sort of intuitive, sort of by nature, I guess, right? Because you have sort of a physical intuition between what the molecules do and I think in the future, right? There's going to be maybe a bit of a merging or a bit of a convergence of these physics-based methods and these fully data-driven methods, right? As I mentioned, right, there's I do think a reasonable claim can be made that many of the like structure prediction or structure design methods learn something akin to an energy, right? Which is a physical property, right? And that can be made in the future. Maybe that can be made a bit more explicit, right? I mean, I think that there hasn't been, as I mentioned, right? The field hasn't been very focused on explainability, right? The field has very much had an engineering mindset, right? We just need to get it to work, right? And I think maybe in the future there's going to be a bit of a shift towards that.
And Stein, is that something similarly that you're that kind of moving from that mindset of we need to get this to work to how do we explain it, how do we interpret it? Is that something that you're seeing in your work?
Yes, it's quite similar in the sense that we work on fundamental problems and want to know how things work. In our case, on the genome, and so one could consider AI models that make a prediction in the clinic to be also explainable to the doctor to kind of know what it works. But for us, it's like only that. So so far we don't really need a lot of models to predict gene expression because we measure it. So it is the whole point. I think for protein structure is still a bit different because you want to design a protein to actually use it. For the DNA also actually we start using that. Or maybe there are similarities and differences. But in any case, the model, when the model predicts gene expression or transcription factor binding accurately, we really want to use the full spectrum of methods to get the explanations out to gain biological insight. And so what I see is that computational biologists are really doing experiments on the model. All this, a little bit like genetics. So we would go into the lab, do an experiment, do the next experiment, try to think, do a next experiment. Here, to me, it's similar. The training the model is somehow challenging, but it's not even half the time or the resources spent in our daily research. Once the model is there, then actually most of the work is trying to use this model as a tool to get to some new insights. And so it's a particular way of working, I think, where computational biologists really have to think about experiments, about controls, all of this while training new models, testing the models, exploiting the models. So it's really fascinating. And for the regulatory genome, so I've been working on this since my PhD when the human genome came out, and with always small increases in what we could understand. And I think with many of us here, we are not here because of a hype or something. It's like really every day. Your talk also was like surprised and amazed by the performances of these things. So at the moment that we were using the convolutional neural networks, the amount of insight we got per day or per PhD student was just a lot higher. And the accuracies are are unbelievable sometimes. And so that is very, very encouraging. And that's why I think we're still doing this. We might think that, I'm still thinking that this is a transition phase, that these black box models are now tedious to decipher and so on. And that they will be very useful to go back to first principles and to exploit these models to get to the actual rules. That is what I always thought, but this period is really lasting quite long, and we are still reaping the fruits of the black box models every day. So as long as we can get new information from it, I think we'll keep going for a little while.
Yes, I did have a moment when you kind of put up the chicken genomes that you were going to unravel. Was at Andre's black box pigeon this morning. So
that was
closing the loop of the day.
That was a beautiful moment today, the pigeon.
I mean, I think because that touches on something I think that you mentioned, Philip, in your talk, which was about this kind of speed and scale of progress. And I suppose there are. I'd love to hear from kind of the panel about what do you see as being the real opportunities for kind of biology over the next 10 years in terms of that, but also what are the challenges in terms of how we make best use of them, make responsible use of them, get the most societal benefit, I suppose, from those. I can go back, unless anybody wants to jump in particularly, I can go back down the panel again, but it's,
Yeah, it's good. To us, it's not the time it takes to build a model. Like you said, we get insights daily, right? And we discover so many new things and get models with great potential. But then validating this model and showing efficacy first retrospectively, and then doing the analytical validation, and then doing prospective validation in clinical trials. So getting these models to the patients takes a very long time today. And I understand why the safeguards need to be in place, right? But on the other hand, it's also an obstacle, it's slowing down the progress in this area significantly.
Yeah, I think the volume of data we have already, it's huge, right? And I'm not sure if we have explored this data at the fully potential. So I see this as a big opportunity for, going deep down in the biology and understanding mechanisms that we have no idea that happen. Right now with the possibility of also using those LMM models to understand how molecules interconnect in the cell, I think this is an open field, and there are some works on this that, they're fascinating. And in terms of health per se, I think we are going towards a period where everything's going to be so connected. And as we saw this morning, instead of maybe treating diseases, we are going to be more at the side of preventing diseases, because I think with this power of AI, prediction is going to be everything, right? So yeah, I'm very excited about what is to come. So.
Actually, yeah, maybe one point to all the young people in the audience, and I see some of you. So I think this is really one of the most exciting times to be doing a PhD, or to be doing research like down at the front, right? And I think it's really, I think you guys are in a very lucky situation, I think, right? I think this is an exciting time. This is something that we always wanted to do, right? But we never could, right? When I did my PhD, we had to do sort of the slow trudging sort of research, right? And you get to do a lot of the exciting stuff. So I think I just wanted to make that point just for all the young people. So in terms of impact and for the near or the medium term future, right? So on the molecular protein side, right? it feels like that things are going to mature, I think, in the next few years, right? And so then from the application of machine learning or computation side, I think it's going to go back to be getting a more systems level understanding, right? I mean, so when I was doing my PhD, when I was doing my postdoc, right, this word systems biology was really en vogue when everybody went to systems biology, right? This sort of died down a bit, right? And I think it's part of the reason was that the understanding we could get from building larger systems level models was just not there, right? It was just too early to do that, right? We didn't know what most of the protein looked like. We didn't know how they interact, right? We didn't know the engineering relation really, right? So I think with so much great work going on now on sort of the wiring piece, on the gene relation piece, on the protein interaction piece, right? I think soon it'll be time to do to build system level models, but then for real, right? And things that actually have predictive power, right? Which we never could in like the 2000s, right?
If I may add to that, so we already see how general AI models like language models and vision models and audio models are starting to integrate and converge right into one unified model or one interconnected model. So I see the same path will follow in our domain, right, where we connect these different layers of biology into a systemic model.
Yeah, I also agree, and I think this is a nice opportunity for our field because let's say in a wet lab environment, consider neuroscience, it is not easy to work both on gene regulation and on proteins and on cells and on electrophysiology, but also on brain regions, on the connectome and on MRI and EEG. It's nobody would be able to bridge these gaps experimentally. And so, but from the data and through models, I think this is, for a computational field, very interesting to actually start thinking about bridging these scales and do causal inferences over these scales, which an experimentalist has much more difficulty, I think, to actually tackle by an experiment. So this will be a really important phase for AI.
Yeah, this will be a lot of fun.
It will be a lot of fun, yeah.
Right, just picking up on the data question, I suppose. I saw a comment on the Nobel the other day about how, essentially that was that was enabled by open, high quality, standardized data sets. Essentially, that was the point. And I think because Mariana, you mentioned as well the kind of the volume of data. And I suppose, like, do you see is that question of the ongoing availability of data a kind of challenge? Are there challenges associated with data, or is the kind of application of machine learning, deep learning tools to data presenting new challenges? Do you think in terms of?
I mean, so I have a one perspective on this. Now, having actively worked in the biotech industry for a couple of years, is that there's a lot of data out there on different properties of antibodies, of drugs, right? And there's but the vast majority of the data sits in companies, right? And these companies have zero incentive to make it open. There's a lot of disincentive, right? Because there's IP considerations and all these things, right? And so there has been always efforts to try to get this data somehow unified and available and make available and then be able to train models, right? As far as I know, that hasn't really happened yet, right? Because there's so many hurdles, right? There's so many issues, right? Once you get to talk to these all these different players, right? But so that is so the simple issue of data availability is I think that is a huge issue. I think, and when you talk about stuff that's going to be commercially valuable, right? Which is a lot of it.
Yeah, I tend to agree with you. I mean, even though in public data sets we still have a large amount of data, some of them are very restricted to big pharma companies and so on. But maybe I would say that the biggest challenge here is lack of metadata and lack of homogenization of the data, because yeah, without metadata, the data sometimes doesn't make sense. And yeah, and it's, I know that for some metadata, we need to protect people's, like for sensitive data, we need to have this special care, how we share the data. But in our community, in general, people don't like, they kind of protect their data. They don't want to share this metadata. So this is a practice that I think we should change somehow. I don't know.
You opened a can of worms with the data question, you know. So I completely agree with you that the data is messy and it's noisy, and we invest, I think 80% of our time goes into data curation, just cleaning up data sets before we can model. But also to previous point, data is siloed in pharmaceutical companies, also in hospitals, especially in EU, where you have different regulations in different countries. It's so hard to get access to medical data. I can give you one example from Slovenia. So we have a lung clinic, right, dedicated lung clinic that treats all Slovenian lung cancer patients in one place. So they have this great data collection because homogeneous population, standardized practice, and so on. They collect RNA-seq, but it's so hard to get access to that data to the extent it doesn't benefit the patient anymore. So I understand those rules, right, GDPR, and other European legislation is here to protect the patient, but at some point, patients are not getting the benefits if this data would be used for research.
Stein, did you want to comment on the data? Or you...
Well, just as an example I was thinking about today is that we've been usually doing model organism research and so on, but our recent investigations on the human genome have, of course, confronted us with questions about how much other data is available for use for training, and how do we share our data, and what do the models contain. So our models that I showed at the end of my talk were trained on human data, all post-mortem, so GDPR is not a problem there. But it's still sensitive, and we have gotten used to data, human sequence data from genomes, to protect it at EGA, to put under controlled access, so that people who want to use it can use it, but there's a data access agreement, and we all got used to that. And now comes the model era, where so we can put the raw data in protected, but then release the model. But like Tim Hubbard was showing this morning, I think it was very nice, I think, you cannot really tell what is in the model. The model may be able to regenerate, given a start of a personal genome, may be able to fill the rest in, and actually this way you can reconstruct somebody's personal genome. And so these things have not been solved yet, and now that we are starting also a new center, I'm thinking, if everybody has to solve these things by themselves, it's going to be a misery. So it's good that Tim and ELIXIR and start to work on this, but it would be great to have some more shared facilities in Europe, like what EBI is doing on data service, that there is additional guidance or infrastructure to make this happen. I think that is what we are now, at least what we are waiting for a little bit. So that we don't have to reinvent the responsible AI and data all individually. It will never work. So yeah. The field is going, the research is going faster, of course, than the infrastructure and then the legislation. So then.
No, it feels like there's always been a kind of arms race, in a sense, about the ability to de-identify data versus the ability to re-identify data. And it feels, after Tim's talk as well, that there's, the parameters of that race have shifted slightly? I mean, is it something that you're encountering as you're trying to do your work, that you're kind of the bounds of like when it is that you'll have concerns about privacy or data disclosure are changing, or?
No, yeah, I think this is something that we have to accommodate all the time. I just received, for instance, from one of our funders that I have to put some, like the consent, how do I say this, the consent that patients, they have to sign to participate in the research. I have to state a specific phrase there, and I have to now, store the data in some specific security requirements. So this, I think, as he said, I think we are learning the way, and we have to accommodate everything on demand, because although I think we can anticipate some problems, as we are saying here, things are happening so faster that it's hard to anticipate everything. So we have to adjust all the time, right?
I suppose actually to go back to the kind of like how you, where the kind of skills and resource comes from for that, I suppose, because you mentioned ELIXIR and things. Do you feel that, you know, as researchers, often we say, researchers should be kind of responsible for all of this, As an ethicist, I tend to kind of walk around a lot going, researchers need to be more responsible. But do you feel that there is that need for kind of new, so obviously there's a huge number of guidelines and guidance around trustworthy AI, responsible AI, but do you feel that there's kind of a gap around biology and around kind of biomedicine, or do you feel that we're just kind of refining? Is it a gap, or kind of is that gap closing?
I think the gap is, I feel that there is a gap. If I compare with animal research, for example, we can do mouse experiments in the lab, and we send in, files to the ethical committee to perform these experiments. But as an experimentalist, we have to go through continuous training of the use and legislation of animal experiments. But that, and we hate that, right? It's like really misery to do that. But it's still there, and so we have always all these additional things we have to do as researchers that take us away from the daily research. It's true, but on the other hand, a minimal set of a minimal structure in our place is actually not really in place. So the ethical committees they don't know how to deal with AI models yet. There is no guidelines to follow for groups. You want to do AI research, then this is the way that you can send in your files. For ERC grants, we have to submit an ethical AI file as well, so that it is starting, and then makes you think about this. But in the institutes, I don't think this is really commonplace to have training and ethical committees being there to assist researchers in follow-up responsible AI. So at least in Belgium, this is not really completely implemented. While for animal research, it is really solid and like over the top almost. But yeah, so we may take examples, the good things there that might be useful, because it forms a guide. I think there's teaching, there's certificates that people get, to work with mice and so on. Not that we need that for AI, but there's a whole infrastructure and I think in the AI research there is almost nothing.
I completely agree with you, and I can share that in Brazil we are also discussing this, but there is no formal regulation about what to do, how to do. So, yeah, we are still talking.
Emerging area. I am going to open up in a minute to for kind of broader questions as well, rather than me hogging them all. But I wanted to come back actually to one of the reasons that I co-opted you onto the panel, Mariana, which is that because in your kind of conversation around ethics and as well, and I think a couple of other speakers mentioned questions of data bias and the kind of representativeness of data, and I suppose, I mean, and that is something that kind of comes up repeatedly as a question, and whether it's something that you feel that we're making progress on or finding better approaches to, or whether it's just something that, you know, is it something that people talk about and then kind of walk away from? And
yeah, I think we are more aware of this bias, but not necessarily tackling this bias, because for avoiding them, we need representative, right, diversity, and as I mentioned, and now I'm talking specifically about omics data, most of people that are represented in those data banks, they are either Europeans or North Americans, so there are lots of populations that are not represented there. So if we are using those, this kind of data to build models and to represent our population, we are aware, of course, that we are not representing tons of people in the planet, right? Yeah, I think, as I said, I think we are more aware, but not necessarily tackling this problem. And another thing I see every time that someone's building a model, people are very concerned about what the model is doing, how the model is performing in terms of assertiveness, in terms of if the model is correct or not, but it's not taking consideration what the model is doing wrong. I mean, is this model, for instance, predicting, I don't know, not recognizing a specific face, or is this model not considering a specific ethnic group? So I think this should be an exercise when we are also building those models, to also pay attention to what we are skipping, what is missing, right? Not only what we are doing great, but also what we are missing.
Miha, is that something that you...
Yeah, absolutely. So diversity is extremely important to us. We want to make sure our models perform well across diverse demographics. So we made it a standard practice to investigate model performance across these confounders like gender and ethnicity, and just the standard pipeline that we always look at that. And we invest a lot in forming partnership across the globe. So we have partners in East Asia, South Asia, Middle East, North America, but then we also realize we don't cover all ethnicities equally, and we are in discussions with cancer centers in underserved populations as well.
Yeah, we do, as molecular people, we don't deal with that a lot, right? I mean, we build protein models, right? So, and I mean, I suppose we could deal with it in the future in terms of, you know, targeting certain variants, right? But right now we target a target, right? And that's supposed to be universal.
It's interesting, kind of like, I suppose, at what point, because obviously, and actually you've kind of almost sat in like a kind of translational pathway, yeah, perhaps, by complete, but I suppose it's that question of like, when do all of these things that we put under this banner of responsible AI or ethical research, when do they become issues for different research areas at different times? I think that's a kind of, to me anyway, it's an interesting question. But I am going to open up to the room as well for questions. Apologies if I can't see you because the light is straight in my face. So, so if you're, but I think John,
So like everyone else, you're suffering with data curation because no one wants to and it's hard. But I don't work for EBI, so I'm just going to throw them under the bus. But if you had one sort of wish from EBI or a large data center, I mean, A, do you think it's practical that they could provide data to cut down on that 80% time? And if so, what sort of thing do you want them to do?
Is that for anybody?
That's for anybody in the room.
Anybody want to volunteer before I just being aware that for the previous questions I've gone that way along the table. So
Do we think we can have standardized general curated data sets? Because each sample is different. You try and do different things. Would that even work?
I have no clue, because then you could limitate creativity and new discoveries. On the other hand, you could facilitate one thing that, so yeah, maybe it could be like, yeah, maybe if someone wants to use the homogenized data, this would be helpful, but also have access to raw data in a controlled way would also help people who wants to be more creative and do different things.
Is it more useful? Because you get data from heterogeneous sites and bring it all together. Is that the problem? Would it be useful for someone to do that?
Absolutely. So I think there's extreme value in standardization of metadata, also of library prep, or the whole process from working with, from the sample to the data, right, in the lab. We've seen some great examples, and that is possible. Just if you look at the ensemble, right, they managed to standardize gene IDs and put all this effort under one roof. That's a fantastic effort. I think it will require a lot of more resources to standardize other data domains, but I think that's necessary, and that's how we should move in the future.
Yeah, I agree. CBioPortal is a very good example of homogenizing data and fostering new discoveries. But this doesn't substitute the access to raw data, so then people can also apply their own pipelines.
Yeah, but the situation right now in the field is terrible. So I can tell you from the example in Slovenia, we are a small country with, I don't know, a relatively small number of hospitals. All information systems for hospitals were designed by the same company, but all information systems in every hospital is different. So they would have different data schema in every hospital in Slovenia, although it was designed by the same company, and it's a fairly small country.
We have this issue in only one hospital.
Can I talk now? Okay,
oh, yeah,
cool.
Sorry.
I had a question on how we keep academia relevant. If you look at large industry models like LLMs, these are so expensive to train that basically they have to be done by very large organizations. And so how can academia remain in the game when this much money's at play? And how can academia remain flat and equal across countries and groups when it requires this much scale, and in biology in particular?
That's a great question.
Yeah. Well, maybe quickly, I think it's a very relevant point, which has a lot of dimensions. Probably there are the huge models that are trained in big companies. Luckily, so far, still often find their way to academia, either because the output is reproduced, like now with AlphaFold 3. Luckily, academic groups, or the weights are released, or they leak, and lead to LoRa fine-tuning and so on in academia, which requires less computational resources. I was more worried maybe 2 years ago. I think academia is coping, but it is an important thing, because our students and postdocs, or certainly students, they are trained in universities and academia. That's the principle of a PhD student, I think. And so if they work on problems which are like completely out of proportion compared to the companies where such students would be recruited, that creates like a weird situation. So I think merely we have to be able to give the students the capacity to work with the most relevant problems. I think we have to keep that in mind. And on the other hand, I also think that in Europe, there's not so many examples of these large models trained in academia, even though there are some resources, quite some resources in tier 0. So I wonder why this is not working well. So, right? So I think on the one hand, we can rely on the big models from companies, but on the other hand, we should also be able to train them. Even if they require a lot of resources, a couple of those models should be able to no, I think it's weird that we're not able to get there, even though in Europe we have a common supercomputer where I bet if you submit a project to get amount all the GPUs, you could get it, but I don't see it happening a lot. So maybe we need a little more culture there.
Maybe I'll add my perspective.
From the US, from Canada.
From Canada, right? I mean, so first of all, if anybody has any GPUs to donate, we'll gladly take them. So I mean, I'm joking, of course, but I'm joking, but I'm not joking, right? But so in my academic lab, right, we're limited to the Canadian public resources, right? And so you can get, my students can get maybe 4 GPUs at a time to train a model, right? Which is already difficult, right? But I mean, I will say that AlphaFold 2, right, was presumably trained on something like 10-ish GPUs, 10 J100s or something like that. So the AlphaFold 2 training wasn't super expensive, right? AlphaFold 3 was probably trained on 500 or 1,000 GPUs, right? And so that's at a scale where it's I would say it's difficult to achieve as an academic, right? I mean, some people can make it, right? You can get donations or whatever, and you can get these resources. Some academics can do that, right? Most can't. We can't. But I will note that it's still several orders of magnitude away from like GPT-4 or GPT-5 or the really big large language models, right? Which are trained on tens of thousands, hundreds of thousands of GPUs, right? So that's when it's like utterly unachievable. When training this model costs you $100 million or something like that, right? Mm-hmm. So in biology, I think we're probably still, and I feel like we're gonna remain just because of data limitations. We're gonna remain in a world where you can get the resources if you really, really want to, and that if you have good funders, you can get the resources probably, right? But still, it would be nice to get. It would be nice to have more GPUs always.
So the general rule in like AI training is garbage in, garbage out. So how do we really address the reproducibility starting from labs? And how do you think that these new workflows and automation comes into play to solve these issues with reproducibility?
Do you want to start?
So that's definitely a big issue for us. We do enforce good machine learning practices to ensure reproducibility in our processing or analytics workflows. So when you take the same sample and run it through our models 2 times, we guarantee you get the same result of that prediction. But then we also need to ensure reproducibility in replicates and to be accepted for clinical trial use. For example, for our biomarker to be accepted for clinical trial use, we had to do the analytical validation, and we had to show that for the replicates of the same sample, right, we can reproduce and make the same predictions with certain accuracy. I guess at this level, right, when you want to enter the clinic, you need to show reproducibility.
I used to say the same phrase for my students all the time: garbage in, garbage out, because if you have like an input and this input is compatible, like it's in a good shape, then you are going to have an output, right? And the way I see this is that maybe first you have to be aware if the data you have, it's capable of answering your hypothesis, your questions, right? Because none of some data, then they can't answer what you are looking for. And second, I think you should have your hypothesis very clear and what you should expect as a result, and having a ground truth as well. It's a good way of certifying that the model is performing good thing, like what would be expected, right? And 3, I think external validation data sets are totally necessary, and people don't use this as frequent as they should. So.
For me, at the end is still always the experiment to do. So if models can be compared and reproduced, external validation data, orthogonal data can be used, and then I think it's really interesting to do an actual experiment that is designed to challenge the model, and that usually gives for us a lot of confidence when these things then actually work, right? For the proteins as well. And if these design proteins actually work, it's like, okay, seems to be a pretty good model.
That's a good indication.
Yeah, pretty good indication, yeah.
Hello. So I was just wondering, we've talked about explainable AI and responsible AI, and I wondered if you see them as two sides of the same coin. You know, is explainability necessary for responsibility in AI? And as we apply more scrutiny to AI models, are we going to need to be able to dig deeper in them? You know, so if you want to see biases in the models, I assume you'll need more and more intricate explainability. And if you want, and so I guess that links in with using more and more resources. If we can minimize the models in some way, then you'll need more explainable model methods to see what it is that you need to really get at in those models. That was for everyone, sorry.
If you want to tackle that one. So I mean, ultimately, so the kind of question of can you have responsible AI that isn't explainable? You know. Anybody want to tackle that one? God, Philip, you're looking.
I mean, from my perspective, I think at least from my perspective as a sort of in the narrow focus on the protein world, right, is the explainability and responsibility as orthogonal dimensions, I think, right, because from our perspective, right, the responsible aspect comes in through applications, right, and you want to make sure, and then I think I'm signatory on one of these declarations of trying to limit the proteins that we do to things that are helpful to humanity and not harmful, right? And so in that case, in that sense, it's orthogonal, right? So you can be fully non-explainable or very non-explainable, but still try to be responsible, right?
I mean, so moving kind of, I suppose, closer to kind of application and kind of regulatory context, I suppose. Miha, do you kind of, is in that context, is that link between responsibility and explainability more aligned?
I think it depends a lot on the context, like mentioned, right? I guess applications like ChatGPT don't need to explain the mechanics of their models. So what they do is they would present use cases, because I assume use cases are enough for their application. But in the context of patient selection and stratification, explainability is a big part of that. And we need to build models that are explainable. You also mentioned the point that there's a trade-off sometimes between model performance and model explainability. So usually complex models would perform better than simple models, but simple models would be easier to explain. So which model do you take, right? We try to look at this change in performance, and if there is no significant difference, we would always go with the simpler model that is easier to interpret.
I'm just thinking out loud that sometimes we tend to choose the simplest model because it's easier for us to understand, but maybe there are things that we don't understand that are very important, and then we are limiting, the power of the tool to our capacity, not the. So yeah, I don't know. I don't have an opinion on that. I mean. No, I do have. I think, well, we should be as transparent as we can, and I mean, sharing codes. I know that for industry it's something complicated because somehow they're protecting their know-how, but I don't know. I think when it's possible we should share codes, and also, when possible, use all the explainability to understand the code, and also because then we can understand, as I said, what we are missing. If we cannot understand what the machine is done, then we cannot see what's missing, right? And then we are going to assume that this is the right way of going, but not necessarily.
It's interesting. I know there's some evidence in terms of the acceptability of kind of data-driven decisions in other domains, where the kind of people are more willing to accept a wrong decision than one that can't be explained to them. It's a difficult trade-off.
And there was a study, a user experience study, that people are willing to wait longer if they see a progress bar. I think that kind of fits here.
Yeah, a kind of transparency in that sense about what's going on, or the idea that something's going on. Here.
Hi, my question got slightly scooped, but I was going to say my second backup question was, I wonder what the panel's opinion is on following trends, especially in machine learning. There's things like LLMs and architectures that are now very optimized for these sort of inferences mean that everyone starts putting them into their applications. And when it comes to explainability, I wonder whether it's worth, let's say the panel's opinion on whether it's worth striking out into unknown directions or into architectures that aren't as popular, and how that trades off in terms of when you're working in research or when you're working in industry.
I didn't completely understand the question fully, so could you rephrase it a little bit?
I'll perhaps try again. But basically, there are very popular architectures like large language models and transformers, which are achieving very high performance but aren't necessarily very explainable or interpretable. And I'm wondering, when you're looking at it from different points of view, like in research or in industry, how much agency, I suppose, do you have to explore new areas, and is that worth it? Or is it best to follow the trend and integrate these existing architectures?
I mean, speaking as an academic, I think we should always explore new ideas and new things, right? I think that's kind of our job, right? And I will restate again something I just I was saying in my talk, right? Is that there were the advent of diffusion in protein models, right? Was arguably transformative, right? I mean, there's been many generative paradigms before diffusion, right? Like GANs, or the VAEs, the autoencoders, right? And many people have played around with them, and we even in my lab we have done stuff on them, right? But it never really worked, right? Even though a fair bit of stuff got into it, right? But then they had a new thing that wasn't arguably not that different, right? But all of a sudden it's transformative, right? And so I think you can only do that if you start experimenting with this new things, right? I mean, there has been talk, or there has been chatter for a while of things that are gonna supplant diffusion in the setting, or that are gonna supplant transformers again in the LLM setting, right? There's every year or so there's chatter about new stuff. It hasn't happened, right? Obviously, right? But I mean, it may happen, right? And so you can only do that if you keep experimenting, right? So.
Well, I think instead of going like surfing the wave, maybe you should choose the appropriate model for your data set. For instance, when I'm working with a very small data set, it's very hard to apply any neural network, for instance. So I think it's very cool to try new things, but maybe first you should identify models that suit better your data.
Yeah, so we have a few initiatives that to investigate methods you mentioned, like transformers, and we invest in that because those would allow certain kinds of applications that are not possible with the current technology, and we want to explore those. For example, digital patients or digital twins, right? If you have a generative model, you can give the model a clinical study parameters and ask the model to generate or simulate a patient cohort with such properties, right? Things like that. So we are definitely interested in such technologies, but whether or not those would be accepted in clinical practice or how we would present that to the public, I think we're still very far away from this stage.
Yeah, I think we've got time for probably one more question. Yeah.
Sorry.
It's sorted out between yourselves.
Very quick question. Like, just a follow-on on this. Like, there's so many foundational models, like so many trained on almost the same public data that's available. Do you think this trend is going to continue with us? Or do you think people are continuing to take it, moving to the next stage of like doing in silico, fine-tuning those models into their specific data sets and use cases and finding more valuable insights out of this?
In my opinion, you'll see that, so you'll have few companies that build foundation models, right? Or few groups that build foundation models. And everybody else will focus more on the application layer, on how to use these foundation models for their particular use cases. So if there was a foundation model of RNA-seq data, the kind of data we work with, I would happily take that foundation model and apply it to our specific needs. That's one reason, but maybe the second reason is also funding, one that we already mentioned. You can do only so much with limited resources, so.
I guess it's also hard to like kind of get like if you are a company like industry versus like academia, it's harder to like fundraise if you're just like fine tuning on existing models. So what are the incentives there? If are we moving into like, okay, like you mentioned earlier in your presentation, training those foundation models on public as proprietary data, or just moving into like using those in a fine-tuned fashion?
I mean, maybe I'll get to say something about one of my pet peeves. Because foundation models, that's one of my pet peeves. I mean, I think foundation model, that's ultimately it's a marketing term, right? I mean, for foundation model, it's just a model that's been pre-trained. That's a pre-trained model, right? The entire concept of foundation models is nothing more than pre-training is useful, right? I mean, that's right. That's effectively what it is, right? So I think if you're ultimately, it's just a question of how much compute do you need to train your model from scratch, right? If that compute is very large, if that engineering is very large, right? Like, I mean, undoubtedly building a GPT is tons of very smart engineering, tons of compute, right? Rebuilding that is going to be very, very difficult, right? But on most data sets that we deal with in the bio realm, right? Training a protein language model, right? That's with billions of parameters. That you can do that. You can do that. With a reasonable amount of resource, right? If you just put your mind to it, right? That's why I think. Anyway, so that's just my pet peeve.
Okay. Who would like the last word on this? Okay. Or not? Okay, well, in which case, I'm going to say, just join me in saying huge thanks to the panel for wrapping up a great day. So thank you.
Thank you. Thank you.
The bar is open, so please head there. So if you have, I know some of you have still more questions for the panel, so do ask them there. And for the speakers, we'll meet at the registration desk at 6 p.m. for dinner.